How are you? Hello, my wonderful people. Good morning.
Morning,
Morning.
sir.
How are
Good
you?
morning, our favorite duck, our one and only duck.
Oh, it's see you. My ladies, are you there? See your gentleman want to con me. I won't mind him. I won't mind him. Flo, are you there? Philomena,
Yes,
are you
please.
there? Yes.
Yes.
It's not the voice of Nicholas I want to hear. Yeah, it's not that voice I want to hear. All right. I hope you are doing very well.
Oh, by His grace, oh,
Okay.
and you?
I'm doing by God's grace. Thank you very much.
Okay. Yeah.
It's actually raining. It was raining quite much in Cape here, so it
Mm.
took me some time to get to the office. I think maybe it's Sunday. Let's wait for a couple of minutes for folks to join, and then we'll kickstart. Let me forward this thing to you right now before I forget. Let me see. So yesterday I asked my TA to get 5 questions for you. Uh, Let me see. Inbox. Um, okay. All right. Okay. So I'm going to forward the questions to you directly. Uh, let's see. Ajuman, Ajingo. I'm going to show. All right. Good. Okay. Ajingo.
Yes, bro.
I beg. I send this thing. I send the trial questions. So please to you right now. So please download it and put it in your group. Okay. All right.
Email.
So yes, I just sent it.
All
I
right.
just forwarded it to you. So please download it and send so that
All
some
right, sir.
nice traffic. Yeah.
Okay.
I've not actually gone through that much, but I guess it's okay. Okay, so
Doctor, I'm here.
Good morning, Doc.
good
Everybody,
morning. How are you?
I'm fine, thank you, Doc. And you?
I'm doing well by God's grace. Thank you.
Dr. Machin, I'm here.
Oh, yeah, yeah, good. Massa, we paid you question that. Massa, we paid you question this morning that. We quiz. You make her right this morning, disgrace myself like that. You see, eh, Massa, why?
Doctor, speak Turkey. Yes,
Massa,
yeah.
we paid you question. Where you make her, you I feel like that. Eh?
Ajibade want you. Doctor, speak Turkey.
Ajin, Ajingo,
I know
Ajingo,
you.
please make sure you. Ajingo, please, have you downloaded it? Please send to the class right now, please. All right, so
Yes,
that
I'm sending it.
so I think I didn't see much of soil water relations. So maybe I will get some 20. I will tell him to add about 20 on soil water relation. Okay, and
Okay.
then yeah, I think I'm just
Okay.
going through the questions right now. I can't see much on soil water. I mean, but the questions are not bad. I think it's something that will be of much help. So I'm going through right now. Not bad, but I think we don't have much on soil water. So
100%.
yeah, it's not bad. I think
Yeah.
that will be enough for quizzes and end of sem once you go through. So I'll add just 20 on soil water. Okay.
Okay. All right.
So all right, that was
So documents. Voila. Here we go.
okay.
Yeah, too.
Hey, the mothers that too. The mothers are in trouble this morning. All right, so maybe just a few people joining, maybe three more people, and then we'll kickstart. Okay. All right. A little bit of
Auntie
yesterday's,
Julie.
a little bit of yesterday's stuff. All right.
Yes,
So,
please.
uh, Auntie
off
Julie,
your mic, Auntie Julie.
your mic is still on. My gentleman could hear the
Yes,
instruction
please.
to the battalions. Eh? They could hear the instructions. Eh? All right. Well, let me see. Let me see. I could. Okay. Let's see. Let me do something quick before we start. Okay, class. Okay. Let's see. I can. Um. Okay. So let's see. Let me see here.
Hello, are you there?
Yes, I'm typing some questions on the screen so that you could just try your hands on it. Please, can you see it? Or you can't. So I'm typing some true or
we
false
can see
questions.
it.
Yeah, so please just try your hand on it.
Doc,
Please,
please, you can see.
yes, please, the ladies, my ladies, please press the neck of this young man for me, okay? They'll be making noise in my ears, so please show them something. Show them something for me, please, with this question. All right, please.
Hello, please. I've sent it onto the biology platform. So when we are done, you can go and download it from there.
Okay, great. So you have 2 minutes more to wrap it up. You have 2 minutes to wrap the questions up. Two minutes more to wrap it up. Two minutes more. Okay. Okay. One minute more to stop work. Okay, stop work. All right, now we are going to mark. Okay, stop work. We are going to do the marking. Okay, so you are starting. Question number 1. Question number 1. What is the answer? True or false?
False.
False.
Okay,
False.
good. All right. Question number 2.
True.
Question number
True.
2. Question number 2 is true. Question number 3.
True.
True.
This one is false.
False.
Because water plants. Okay, water plants. Those are some of the water plants. Some aquatic plants don't have stomata. All right, let's see. A high concentration of carbon dioxide in the intercellular space of the leaf. Okay.
It's false.
Induces stomata.
False.
False. Cyanide can inhibit ATP production.
True.
True.
That's true. Abscisic acid in the gas cell in the gas cell induces the stomata opening, the opening of the stomata. True or
False.
false?
Correct. False.
That's false. Because it will prevent the influx of what? Potassium ions. Okay, suction pressure equals tego pressure minus osmotic pressure.
True.
False.
That's
False.
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 Please, those who said true here, I don't want to finish and I want to hear 50 over 50, you know, the men in particular. I don't want to hear that 50 over 50.
False.
So ladies, please make sure you take note. Those people, somebody just said true here, and I won't be surprised if they say 50 over 15.
And the ladies two people say
All right, so do one
true
person.
where it is.
So
The
do
other
one
is
person.
false.
So do one person. Afraid is so do one person. Don't bring yourself. Don't bring yourself. Even that way, it's a slip of tongue.
Is that your man?
And then the total amount of water absorbed is termed as what? Is termed as filled capacity. True or false?
True.
False.
False. That's false. A lot of men are dying. A lot of men are dying. A lot of men. I'm not surprised. Glycolysis is energy. It's an energy
So,
dependent.
sir, is it false or true? The question is.
Which one? The, no,
Question
man,
eight.
that's
Question
false
eight is
statement.
false.
That's false statement.
Okay, false.
Glycolysis is an energy dependent process. True or false?
True.
True. False.
True.
It's true. You see, I heard about
It's
two men
true.
saying it's true.
Man down.
One
One
man
man
down.
down. It's true. One man down.
2 ATP, 2 ATP, 2 ATP.
You see, I'm not talking. No,
Ah.
I am not talking. I'm
And
just watching you.
no, you are not talking because you don't want them to know you are getting them wrong. That's why you are not talking. But we know, you are dying slowly. We know you are dying. Glycolysis is both an isogonic and endogonic process.
True.
True or
True.
false?
It's true.
It's true. Good. Hexokinase catalyzes the conversion of glucose into fructose phosphate. True or false?
False.
It's false. Convert it to what? To glucose 6-phosphate. Glycolysis produces 2 pyruvate, 2 NAD plus, and 4 ATPs. True or false?
False.
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 It's false. It's 2 NADH. 2 is 2
Down.
NADH. One man is down. It's not DH plus. Good. It's not D plus. Good. The cleavage of fructose 1,3-phosphate is catalyzed by aldolase. True or false?
True.
True.
It's 1,6. It's not 1,3. I'm sorry, my friend. So
One lady is down.
only one lady is down, but one man is down as well. So it's false. It should be 1,6. It's not 1,3, please. And then glycolysis is exhibited only by plant and bacterial cells. True or false? That's false. By almost all living organisms. And then the last one, 3 main processes of cellular respiration are photosynthesis, glycolysis, and electron transport chain. True or false?
False.
False.
Is glycolysis,
False.
Krebs cycle, and electron? All right. Okay.
100% false.
Who are 15 over 15?
Number 6. I didn't hear. My network was off. Number 6.
Too wrong.
Number six is false.
Okay, thank you.
Actually, I say. Okay. Now, who are 15 over 15? Let me come here. Who are 15 over 15?
I don't think.
Too wrong.
Who are 15 over 15? Yes. Isaac. Oh, Isaac, well done. 11 over 15 is not bad. Good. That's a good score. Okay, who had 15 over 15? Please, I don't want to see any man typing 15 over 15 here because we all Belinda, well done. 13 over 15. That's very good score. Excellent. Proud of you. Proud of you. Belinda, proud of you. Ajiman, I want to see your score. Ajiman, Emmanuel, the two Emmanuel, Ajiman. Please, Ajiman has suddenly disappeared. Oh, Anita, well done. Erico 10, not bad, not bad, not bad. Ajiman, okay, Iman, Aponsa, well done. 13 is not bad. Where is Ajiman? The man has disappeared. He will come back to tell us. Come back to tell us 15 or 15. It will be war here. War unto him. We are waiting for him. Let him come back and tell us 15 or 15. Yes, yeah, Iman, well done. Iman Aponsa, well done. 15 or 13 or 15. That's very good score. Please, yes, who else again? Hey, where there is silence in heaven, what is happening? Did people die? Yes. Oh, okay. Sammy, not bad. Yes, Sammy, 11, not bad. Okay. Franco, 18. That's good score. That's very good. That's very good. Okay, Bashiru. Hey, Diana, King Bashiru. 12. That's a very good one. That's very good one. Okay, Prosper. Okay. Julie, good. Anna, 11, not bad. Not bad. Owusu, no, that's good. 12. That's good. That's good. Prosper, 12. That's good. Yeah, Paul, 11. That's good. Where is that man? This man will come back to tell us he had 15. I'm waiting for him. Let him come back. Oh, Lord the Kingsley, 12. Not bad. Nico, 11. That's good stuff. That's very good. That's very good. Okay, these are very good revision questions. So please, make sure you take screenshots of them. Okay. TJ, your hand is up. What did you get? Philo, I've not seen your score. Tell me something sweet. Yes, tell me.
Please, I had 13.
Oh, that's good. Oh, TJ, 12. Okay, Philo, 13. That's a very good score. Well done. TJ, what you get?
I had 12.
TJ, so
I had 12.
is your
Yes, sure.
hey,
My heart says out of 15.
we need pink sheet. Pink sheet. But today your man Ajiman, he disappear. As we start the mention score, though, the man disappear.
He will come back. He will come back.
Make he can't tell we say get 15.
He get 15.
His network.
All right, okay. Yeah. Alex, 12 is good. Ernesto, 11, not bad. That's good score. That's good. That's good score. That's very good. Ernesto, that's very good. Yes. The ladies, my ladies, where are you? Please. Yes, I can see the ladies. Where are you? I can only see Anna, Juliet, and Anita, Belinda. And what are my ladies? My ladies, tell me something sweet. Let me see your scores. Yes. Where are my ladies? Hey, the men, have you stopped typing your scores? Ajim, what did you get?
So I had 13.
13.
13. Honest speaking, I don't lie. I'm a man of God. I don't lie. I had 13.
Ajim, you don't know how boys they wait you for here. All right, 13 is not bad. We're waiting for
because I'm so special.
We're waiting for you to come and tell us. We're waiting for you to come and tell us.
A lot of
15. Okay.
Oh no, I had 13. Honestly, I had 13.
Okay, that's good. That's good. Yes.
But the power said in me, yes. I will remove you from this platform.
Oh, the man. Okay, well, Owusi says Ajungo did the line, but well, it's Sunday, so we'll trust Ajungo's ball, but with a pinch of salt. You know what I'm saying? You know what I'm saying?
The power vested in
Oh,
me as of
Victor,
course,
Victor,
I
what
will
is your
remove from this
score?
platform.
Victor, what is your score? Yes, Victor. Mm-hmm.
Doctor, I
Okay,
scored 13.5 because
look,
of
Yadwell,
media. That's why I don't want to mention my score.
look, I need taking Lotto.
The question
Yadwell.
12.
Oh,
I
you
had
didn't see the D plus. Oh,
I
sorry
didn't see the
about
D
that.
plus.
Oh, sorry about that. Oh, Messi, my two
Uh,
Messi. Where are you? Messi, a child Messi, now, please. And Messi, Fiati, please. All my three Messis. I want to hear your scores.
Doctor, I got 10
Oh,
over
that's
15.
good. That's good. That's good. That's good. That's good. That's good. Valencia, please, your score. Eh? Rosemond. Rosie. Rosie, tell me something sweet. Tell me that you had 14. Tell me something sweet.
God, please, I have 13.
Oh, that's good. That's good. That's good. Praise. What did you get? Praise. Oscar, I won't hear your score. Massa, I won't hear yours. You see, the men, when they don't get anything below 10, they need to Joshua, I won't see your score. Joe, hey, Johnny, I won't see your score. Afiz, King Afiz, what did you get? King Afiz. Okay, Joe, 10 not bad, 10 not bad. My two gifties. Oh, where are you? What is your score? Sisi, Princess Cecilia. Please, what is your score? Asha, what did you get? Anthony has been silent. Yes, Anthony. Oh, Valencia didn't take part. Oh, okay. All right. But please take a screenshot of the questions, okay? Because they are nice revision questions. Okay. All right. I think you guys have done very well. Okay. All right. Okay. I think not bad at all. That's a very good score. Well done. Okay. So we are going to talk about what? We are going to talk about the second half of our lecture today. We are going to talk about the second half of our lecture today, but I think you have done very well. And next to 11, not bad at all. Hey, Kina fees 12, CC 12. That's good. That's good score. Well done. Okay, that's good. All right. So yesterday we were able to let me pick a new slide. Let me pick a new slide here. Okay. So yesterday we talked about what? We talked about glycolysis. Okay. We started with a cellular process. Okay. That's cellular respiration. We started with the cellular respiration, and we said that there are three main processes. What are they? Glycolysis. All right. Glycolysis to what? To Krebs cycle, Krebs cycle to electron transport chain. Okay. Electron transport chain. And then we said that glycolysis, well, is what? It's ATP dependent process, requires 2 ATP. Okay, so glycolysis involves splitting of what? Glucose to what? To pyruvate. To pyruvate plus what? 2 NADH, and then plus 4 ATP. All right, but remember, glycolysis is to require 2 ATP. All right, and because you require ATP at the end of the process, the next word ATP is equal to what? 2, because you spend 2 to get what? 4. And then we know that it doesn't, okay, it does not require oxygen, and then it occurs in the cytoplasm. All right, great. Okay, so basically this is what we talked about, and then we also say that glycolysis requires what? Glycolysis requires what? NAD plus to be present. Okay, NAD plus to be present. Okay, NAD plus. So it is that what will be converted into this. Okay, NAD plus to be present. All right, good. All right, so this is what basically this is our recap of yesterday. Now, at the end of the then we said that the first phase, the first step of glycolysis, the first step in what? Glycolysis. Okay, glycolysis is the what? Is the phosphorylation of what? Phosphorylation of what? Of glucose. Of glucose by the enzyme called hexokinase. Okay, and basically this one is what? Is to pin the what? The glucose down. Okay, so because they will add the what? The phosphate to what? To glucose to give what? Glucose 6 phosphate, so that it cannot go back the way it came.
Please, Doc.
Yep.
That's so you made it is
Oh, so that so
no back
okay.
behind the that.
So that
Yeah.
it cannot go back the way it came. All right, great. So this is basically what happened yesterday, and then we know what happened. We get a 2-pyruvate at the end. Beautiful. Now, so today we are going to take the second step. The second step is what? The second step is the what? Is the now after glycolysis, okay? After glycolysis, okay, glycolysis, okay, where we've had what? Next ATP of what? Next ATP of 2, and then 2 NADH produce 2 pyruvate. And remember, pyruvate is what? 3 carbon molecule. Glucose is what? Glucose is 6, so they will split the glucose into what? Into 3 carbon what? Molecule. Beautiful. Now, with this glycolysis, when oxygen is present, okay, when oxygen is present, when oxygen, okay, so at the end of glycolysis, okay, remember glycolysis is what? It's exhibited. It is what? It is exhibited exhibited by what? By almost all living organisms. All right, good. To extend that, even if glucose is not present, they wouldn't mind to use glycerol, which is fat, which is product of fat, or they will use what? They will use alanine, which is what? Amino acid from what? From protein. So that tells you how glycolysis is very, 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 very important. Okay, so now at the end of glycolysis, okay, at the end of glycolysis, okay, we've gotten our glucose, okay, plus what? Plus NAD plus 2 ATP. All right, like that. So we've gotten this. Now, at the end of glycolysis, we've gotten this beautiful thing. So what is going to happen? Now, remember, all this one is occurring in the what? It's occurring in the cytoplasm. All right, it's occurring in the cytoplasm. Now, once I'm done with this one, the thing is that if this is occurring in the cytoplasm, then when oxygen is present, then a certain process we call what? Transitional what? Reaction, okay, will take place. All right, so that is on condition that oxygen is present. So if oxygen is present, then a transitional reaction is going to take place. And what is the transitional reaction? When oxygen is present, then the pyruvate, the two pyruvates, okay, will now be converted. Okay, now remember I told you that pyruvate is what? It's a three carbon molecule, right? Glucose is what? Glucose is 4 carbon molecule. Okay, so this pyruvate here, please listen to what I say. When oxygen is present, because oxygen is not present, this reaction is not going to occur. So when oxygen is present, this pyruvate will be converted, okay? Will be converted into what? The transitional process will occur. And this transitional process will occur, converting what? This pyruvate into something we call acetyl CoA. And this acetyl here, okay, this acetyl here is what? It's a two carbon molecule. The acetyl here is what? It's a 2 carbon what? It's a 2 carbon. Good morning, how are you? Good. It's a 2 carbon what? It's a 2 carbon molecule. All right. Now, so the question I'll ask you is that, okay, if this is a 2 carbon molecule, which molecule is going to be, what has been removed here? Remember, I just said oxygen is present. So who can tell me? So which means that a certain molecule has been removed. which molecule will be removed? Yes. Somebody should help me. Yes. What will be the molecule here? Let's see if I put something like B here. What is B? Yes. What is B? Yes. Yeah. What is B?
Doc,
Hydrogen.
Yep. No.
is the NAD?
Is there? No. Yes.
Carbon.
Carbon. Carbon has, but oxygen is there with the carbon. So can you mention the molecule? The molecule that will be coming here. Remember, I just said if oxygen is present.
So carbon dioxide.
Carbon dioxide. Only lady. Only lady. Oh God. This class now, the ladies in this class, they are something else. They are too brilliant. Why? A lady could say this. Okay, good.
Hey.
Okay, so here, this process, the removal of carbon. So you can see that this acetyl here is a 2-carbon molecule. Acetyl is what? It's a 2-carbon molecule. All right. So when oxygen is present, please don't forget this. Don't forget this. I love questions in this area. When oxygen is present, that is when this transition step is going to occur. And that transition is just a transient, just a short step. Okay, it's a short step. Okay, that's going, and that short step is what? It's a removal of what? It's a removal of carbon. Okay, and by that removal of carbon will be in the form of what? Carbon dioxide. And when you remove carbon, okay, what will be the name of the process? This process is called what? So this process will be called what? Decarboxylation. Decarboxylation. All right. Okay. Decarboxylation. Are you with me? Are you there?
Yes, Doc.
Yes. Decarboxylation. All right. And what will be the name of the enzyme? What will be the name of the enzyme? So the enzyme for this, so removal of the carbon here, okay, remember it's 3-carbon molecule. Now we've gotten this 2-carbon molecule, which is the acetyl, okay, and then they will add the CoA to it, all right, okay, the CoA will ensure its transport to the what? It's transport into the Krebs cycle, okay. So this carbon here, so we involve the removal of what? So oxygen is now present, so we get what? So this is what? So this is what? Decarboxylation. And the enzyme for this decarboxylation is called what? It's called pyruvate dehydrogenase. Okay? So the enzyme here, the enzyme for this what? Is called pyruvate dehydrogenase. Okay? All right. Okay? So decarboxylation process is going to occur, all right, and this decarboxylation process, okay, all right. So, and you know what? It is not only carbon dioxide that will release. The interesting thing is that another substance, another substance here, NADH, will also be produced. All right. Okay. Now remember, I told you that how many molecules of pyruvate will be produced in glycolysis? How many molecules of pyruvate will be produced in glycolysis, please? Yes.
Two.
All right. 2 pyruvate here, right? 2 pyruvate will be produced in glycolysis, right? So you can see that every process that I've written here, everything here will be what? Will be ×2. Have you seen it? Because the pyruvate here, we have 2 pyruvate what? We have 2 pyruvate molecules. Okay, so everything I've written here will be ×2. So which means that because 2 pyruvate is here, when oxygen is present, we will have what? 2 acetyl OA. OA will be produced plus what? Plus 2 molecules of what? Carbon dioxide plus what? Plus 2 molecules of what? Plus 2 molecules of NADH. Please, are we clear with that? Hello?
Hello, sir. Please. For
Should I
the
come again?
acetyl,
Yes.
for
So
the
let's see.
acetyl CoA
Yes,
is 1.
Yeah, so no, so here is 1. Look at here. You see this process, everything here is 1, right? But remember, no, it's 2, so I'll put 2 everywhere. So 2 here, 2 here.
What about the COA? I'm talking about
No,
CoA.
they are together. They are together.
Yeah, okay.
They are together. Yes, they are together. Very good. So you can see that the pyruvate produced, because 2 pyruvate will be produced, 1 pyruvate will undergo this reaction. Like that. Okay? And this reaction, remember, is occurring in presence of what? When oxygen is what? So here, when oxygen is present. If oxygen is not present, this reaction is not going to occur. Okay? And this reaction, when it occurs, what are we going to get? We will get acetyl CoA, we will get carbon dioxide, and we will get what? We will get NADH. 1 NADH will be produced. Okay? But we have 2 pyruvate are there. We have 2 pyruvate. And so if we have 2 pyruvate, then everything here is times what? Times 2. Okay? Everything here will be times 2. All right. So which means that at the end of the word, so the transition process will end, we'll have each pyruvate undergoing what? Decarboxylation. Which
Hello,
is
Doc.
catalyzed. Hello, please. Go ahead.
Please, if it is occurring in the presence of oxygen, why are we not adding the oxygen to the pyruvate?
No, the oxygen is not really involving directly, but its presence is what is necessitating the process. Do you understand what I'm saying? Let's take it this way, that you are playing football, okay, like, you know, those days, the good old days of JSS, you are playing football. Sometimes you'll be playing by heart, and maybe when you mix some clear chance, your teacher will call you to the side and then say, oh, they will just, when there's like, they will just call you as if they are speaking something to you, your ears, and then you take 2 lashes at the corner. So the 2 lashes you get from the corner, when you go back to the field, you will be playing, because once you know your teacher is at the side door, okay, at the side of the lines with a cane. In fact, you will play as if tomorrow your life depends on it, like oxygen. Do you get it? Or let's say you've gone to a play with your mom, and then a certain child is misbehaving. Sometimes the way your mom would turn and look at you with his eyes, warning you that, Kwame, the day you try this nonsense at home, you like your mom is annoyed because of seeing that child misbehaving. I mean, your mom will not do anything to you, but her presence with that eyes alone, you get it? Okay, so just the presence alone will trigger the process. Do you get the point?
Yes, Doc. Thank you very much.
So you can see that if you look at this one, look at the name of the enzyme. Have you seen the enzyme? You see this process, it is more of removal of hydrogen. Have you seen it? It is oxidation process, but it's not so much of addition of oxygen, but it is more of what? Removal of hydrogen. Remember, oxidation process could occur as a result of what? Removal of hydrogen. So you can see, if you look at the name of the enzyme, dehydrogenase. Okay, and the way D means removing. Do you see it? Are you there?
Yes, it's well taken. Thank you very much.
Good. So here we have the 2 pyruvate molecules giving us the what? Giving us the 2 acetyl CoA, and then what? 2 carbon dioxide and 2 NADH. Because each of them here, this one will produce 1, but we have 2 pyruvate. So at the end of the day, we get 2 acetyl CoA molecules, 2 carbon dioxide molecules, and 2 what? NADH. And as I said, this is decarboxylation process. So after the removal of this one, after the removal of the carbon, then the oxygen will be what? Bonded to it to get our what? Our carbon dioxide. Okay, like that. So we have our what? This is our intermediate process. It's a short process. It's a transient process. Okay, very short one. That's why we call it what? Intermediate. So that's why when we are talking about the cellular, we don't really mention it. Okay, we don't really mention it. Okay, all right. We talk of the glycolysis, we talk of the Krebs cycle, we talk of the electron transport. Okay, but we have this important intermediate process. Okay, that will ensure that with the acetyl CoA, okay, molecule, the energy, the currency that is going into the what? The currency to the to the mitochondria is in right currency. Why? Because we don't need pyruvate. Pyruvate is not the one that will enter the what? The Krebs cycle. Okay, but it is the what? It is the acetyl CoA. So if oxygen is present, this pyruvate needs to be converted into acetyl CoA. All right, good. Now that I've explained that to you, let's see what we have here. All right.
Please, Doc.
So, hello.
Please, Doc. I have a
Hello.
question.
Please go ahead, please.
If you can get back to the
Go
previous
back. Okay.
slide.
Thank you.
Yeah, concerning the dehydrogenase of the pyruvate.
Yes, please.
You said it talks about removal of oxygen. Or
removal of hydrogen.
hydrogen. Okay.
Yes,
Okay.
yes.
Okay. All right. Thank you very
So
much.
it is also oxidation process. You see, remember yesterday we said oxidation can be removal of what? Oxidation can be removal of electron or removal of what? Hydrogen. Okay. Or addition of what? Addition of oxygen. Okay, good. So that is that. So removal of what? Removal of electron or hydrogen. Okay, or addition of what? So that all these what? All these processes are what? Oxidation. Okay, and in this case, you can see even if you look at the name of the enzyme. Okay, and even that is why even we have this as a product. So which means that in this process here, we needed what? NAD plus were there, okay, to pick up the what? To pick up the hydrogen, the removal of the hydrogen. That's why the enzyme catalyzing it is what? We have the what? We have the pyruvate dehydrogenase, okay, pyruvate dehydrogenase, all right, pyruvate dehydrogenase, okay, good. All right, so now let's see, good. So this is what you have in your notes. That's what I've explained here. So you can see here, my pyruvate is here. It's a 3-carbon molecule, all right? And this 3-carbon molecule here, okay, what is it going to do? The removal of what? The remove one carbon from it, and the removal of the what? Of the one carbon, that's a decarboxylation. And you can see that what? We have that our NAD plus, okay. This one should like that. This one here should go. Let me do this one. Okay, so here should go up like this. I'm coming. Here should be like that. Like that. Okay. Okay, good. Are you with me? So this is how the diagram is supposed to be. You get it? So they will remove the what? They will remove when they remove the hydrogen. Somebody, when the hydrogen is removed, somebody should be there to capture it. Okay. All right. So let me put this one. Let me make it this way. All right, like that. Okay. So when they remove the hydrogen here, somebody here should, this guy here is there to capture the hydrogen and form the NADH. All right, good. So look at that like that, very simple. And then the enzyme is what? Pyruvate dehydrogenase. Okay. And then in the process is what? It's decarboxylation. Okay. Decarboxylation, removal of what? Of carbon. Okay. Forming these two molecules, acetyl-CoA. All right. Good. Beautiful. Now, remember, this process is occurring where? In the mitochondria matrix. Okay. All right. In the mitochondria matrix. Okay. In the mitochondria matrix. Next. Okay. Hello. Yes, go ahead.
Please, the acetyl coenzyme A is the same as the pyruvate dehydrogenase, no?
No, pyruvate dehydrogenase is, let me see, and let me pick that. Let me go. Okay, so let me put that one. Oh, look at that. Now, acetyl, the enzyme is what? Pyruvate dehydrogenase. So this is the enzyme. Okay, the acetyl-CoA, this one is organic molecule that will be added later on. But the main thing is acetyl here. Okay, the main thing is acetyl. The acetyl is the two. But this one is we take this one from vitamin B. Okay, we take this one from vitamin B. That will be after the formation of the acetyl, then the coenzyme A here will be added to it. Okay, but the enzyme to catalyze that, this conversion, okay, is the what? Is the pyruvate dehydrogenase. Okay, the enzyme to catalyze that is pyruvate, and you can see one carbon dioxide here. Okay, you can see one carbon dioxide. Okay, here, like that. All right, good. Okay, all right. Now, don't forget. Let me pick image from. Let me pick image online for you for. Let's say. Let me get image online for you for this one quickly. Let's say. Let me pick my. Let me get a detailed image for you here. Big mitochondria. Okay, so let's say, all right, let me get a big one. Okay, let's see. Okay, I think this one is big, a very big one. Just a minute, okay, let's see. Okay, all right. So let me take this one. Mitochondria diagram, very big one. Okay, so let me pick this one. All right, so come here. Oh, mitochondria. Oh, this one that I pick is not coming. Please, just a minute. I want a very good image. Okay. Let me crop it because it is just not allowing me to. So let me crop it quickly. All right. So let me take a picture of it. Pick one like that. Okay. Copy. And then come to my slide. Great. So let's say this is mitochondria. Okay, good. So what's happening is that this intermediate reaction will occur in the what? This is the matrix. The matrix is the liquid portion, okay? And then if you look at the mitochondria, you can see that this part of mitochondria, if you look here, look here, like that. If you look at the mitochondria, there's the liquid part, that's the matrix, okay? And then the infolding is the crystal. That will increase surface area. Okay, all right, great. So it is within this matrix that is where my intermediate process is going to work. The intermediate process. So intermediate, okay, that's where my intermediate process is going to work. The intermediate or transition process, okay, occurs. All right, good. So this is inside here, all right, and the process will occur only when oxygen is present. And then when oxygen is present, then inside the pyruvate will be transported here to the matrix of the mitochondrium, and then it will be converted into what? Into acetyl-CoA by an enzyme called what? Pyruvate dehydrogenase. That will catalyze the removal of what? Of carbon dioxide. And then that is what decarboxylation process. And this will be our product. And remember, there are two in front of every word here, because two pyruvate molecules will be produced at the end of glycolysis, and each of them will undergo the process. Okay, are we there? Any headache so far? All right. Now, at the end of this transitional process, then we are going into the what? Into the Krebs cycle. Now let's look at the Krebs cycle. Okay. Remember all these things that I'm talking about. I said when oxygen is present. So everything that I'm talking about here is when oxygen is present. Okay. So let me even jump this step here. Let me come here. All right. So let's take it this way. Within the cytoplasm, glycolysis has occurred. And the glycolysis occurring in the cytoplasm. Then when oxygen is present, then the pyruvate will be transported to the what? To the mitochondrial matrix. Okay, and inside the mitochondrial matrix, okay, what is going to happen? We are going to change the they are going to change the pyruvate into acetyl-CoA. Okay, and it is the acetyl-CoA that will enter the cycle we call the what? The Krebs cycle that is going to occur in the what? In the mitochondrial matrix. Okay, so let's see. You are going to America. All right, you are going to America, what you do is that, what do you need? So when you get to the airport, what do you do? Or somebody from America, all right, from America has come to Ghana. At the airport, you'd have to what? You have to change the dollars into what? Into cedis. At the airport, you have to change the dollars into what? Into cedis. So that's what you need to do. At the airport, you have to change the what? The dollars into what? Into cedis. Okay? Right. So changing the dollars, and so let's take it this way, that the person coming here, all right, you only change the dollars into cedis only when you are in what? You are at our airport. Okay, if you are in America, and you wouldn't need to change the dollars into cedis because you don't spend cedis in America. You only change the dollars into cedis when you get to the airport here. All right, when you get to the airport here, that is what you do. Okay, so let's take it, the person who has come to Ghana, you are from where? You are from US, then you've come to Ghana. And when you come to Ghana, you are having your dollars. So the dollars is the pyruvate. So the moment, remember, the changing of dollars into cedis is occurring in Ghana. So the cytoplasm is the what? Is the US. And then the moment you come to Ghana, okay, which is inside the mitochondrion, then you have to be converted. The dollars that you do, which is the pyruvate, must be converted into what? Into cedis. So the cedis is the acetyl CoA. All right, beautiful. Now, once you get your acetyl CoA through a transitional process, so the transitional process simply involves the what? The formation of what? The transitional process, the formation of what? Acetyl CoA. And then the other accessory product to carbon dioxide, and what? N2 NADH, like that. Great. Now let's see. So this is how the Krebs cycle is going to be. All right, good. Now let me get the detailed image for you. In fact, I'm not expecting you to put all the, let's say Krebs cycle here. Let me get a simple image cycle for you. Okay, simple image cycle for you. Okay, I'm coming. Let's see. Okay, simple image cycle for you. Okay, let me copy that. Okay. All right, let me bring another one. Another one with enzymes. Okay. All right. So this one is the detailed version. Like that. Okay. All right. Okay. So this is the detailed version. All right. So let me start from here. So this one, overview here. So the acetyl CoA. So here now the Krebs cycle, we know it's what? It's occurring in the what? So inside the what? inside the mito, so within the what? Within the mitochondrial matrix, we know that intermediate reaction has occurred, which is the word, which is the intermediate reaction has occurred, which is the word, which is the acetyl CoA formation. All right. Okay. Then afterward, this acetyl CoA will enter. Okay. Now that you've entered, the acetyl CoA is the currency now. CoA. It's now the currency. That's the coenzyme A. So this is the currency that will enter a cycle. So this is the currency you need to spend in Ghana. So this one is equivalent to our cities here. So now that's why I said here, now you are here. You are inside here. All right. So the Krebs cycle. So after the conversion, then this acetyl CoA will enter the Krebs cycle. Okay, good. So this is quick overview of the Krebs cycle. Now it's a cycle. Okay, so but the acetyl CoA will enter at a specific point. Now, before the acetyl CoA enters the Krebs cycle, have you seen that? You see that the CoA will break off. The CoA part here, the CoA part will break off, and only the 2 carbon molecule here, only these 2 carbon molecule will enter to just break off away. The coenzyme part, okay, the coenzyme A will break off, and only the acetyl, the 2 carbon. Remember, there were 3 carbon under pyruvate. They were reduced to 2 carbon, okay, and then this coenzyme A is actually obtained from vitamin D. Okay, vitamin D was added to it so that it could be transported to to this Krebs cycle. When it is entering the cycle, the CoA breaks off, and this 2 carbon molecule acetyl will enter the cycle. Now, so look at the process. Look at the name here. Now, if you look at the first slide that I showed you here, look at the first slide I showed you. You can see the Krebs cycle is also known as what? Citric acid cycle. Citric acid cycle, which is going to occur in the matrix of the mitochondria. All right. Now, so the citric acid cycle is because you see that the first thing that will be formed is a cycle, but this acetyl will enter at this very point, whereby it's going to react with what? With the oxaloacetate. Okay, oxaloacetate here, which is a 4 carbon molecule like that. Okay, to give us the citrate, the 6 carbon molecule. Okay, so because of this, that citrate, okay, that's the formation of the citrate. Okay, that is why we have that name. That's the citric acid cycle. Okay, so that's another term for what? Krebs cycle. Krebs cycle was actually named after the scientist who the Krebs. His name is called Krebs. It's called Hans Krebs. Okay, that's why it was named after him. Okay, now, so this cycle, what is going to happen? The acetyl will enter the cycle. The first thing is going to is going to react with this 4 carbon molecule. Okay, that's the oxaloacetate to form the citrate. So that is the first and most important step. So remember that this is 2 carbon, and when this 2 carbon comes here, it's going to react with this 4 carbon molecule and then give the citrate. Okay, now look at it, it's a cycle. So you see, when it goes round, it's a cycle. So when it enters and goes round, we end up coming to what? Regenerate our oxaloacetate again. Okay, so it goes through the cycle so that you can see here the oxaloacetate here will react with the what? With the acetyl CoA. The acetyl, the CoA will break up, will react with the acetyl, yeah, okay, to form what? The citrate. And this citrate will enter into the cycle, and then we'll do what? We'll lose 2 carbon, the 6 carbon molecule here, we'll lose 2 carbon dioxide, okay, and then we'll generate the what? The oxaloacetate again, and then the cycle continues. Okay. Now, so let me go the overview again. We have our acetyl CoA. This is the currency. Okay, this is the money, the currency you need. And this currency we need is the what? Is the acetyl CoA. Okay, so this CoA here, when it's about to enter into the cycle, the CoA part is what? Is broken off, so that only 2 carbon, the acetyl group, will enter the what? The Krebs cycle. And the first step in the Krebs cycle is the formation, is the what? Is the reaction between the what? The oxaloacetate. The oxaloacetate, which is a 4 carbon molecule, to form the citrate. That is a 6 carbon molecule. But remember, it is a cycle. So this citrate will undergo a number of what? A number of metabolic processes. All right. And during that process, okay, during those processes, 2 molecules of carbon dioxide will be what? Released. 2 decarboxylation processes will occur. And these 2 carbon dioxide will be what? Will be released. And then we'll get our oxalo what? The oxaloacetate back. Okay, and then the cycle continues. So Krebs cycle will involve reaction of the oxaloacetate to get with the what? With the acetyl group to get the what? The citrate. And then the citrate will undergo reaction to generate our the oxaloacetate back. Now, the question I want to ask you is this. Have you noticed that all the glucose that we started with has gradually disappeared in the form of carbon dioxide? Who has noticed that? Remember, the acetates here, there are 2 of them. What I'm showing you just one. Remember, at the end of the glycolysis, 2 pyruvate, okay, 2 pyruvate molecules were produced. Are you with me? At the end of glycolysis, 2 pyruvate molecules were produced. Are you with me? And these 2 pyruvate molecules were produced means that 2 acetyl CoA were produced during the what? During the transitional phase. 2 acetyl CoA molecules here. You can see from here, 2 acetyl CoA produced in the what? In the transitional phase. So right now, this Krebs cycle I'm showing you, I'm only showing you one here. Okay. So during the what? During the Krebs cycle, 2 decarboxylation reactions will occur. Okay, resulting in the removal of what? 2 carbons in the form of what? Carbon dioxide. All right. Are you with me? Because the oxaloacetate that we had, it has been regenerated, but the 2 carbons that entered the cycle has been, the 2 carbons that entered the cycle have been removed in the form of what? Carbon dioxide. Please, have you noticed that what entered here, if we remove them back, okay, it has been removed because we've gotten our oxaloacetate back. Are you with me? Any confusion? Any confusion here? Any confusion here? The 2, you see, 2 entered here. 2 entered here. 2 carbons enter in the form of what? Acetyl acetyl what group. Acetyl CoA here, but when it's about to enter 22 carbon. And then but at the end of the cycle, we've gotten our oxaloacetate back and 2 carbon dioxide removed. So the 2 carbons that entered here has been removed in the form of carbon dioxide because we've been able to regenerate our oxaloacetate. Okay, we've been able to regenerate it.
So doc.
Yep.
So what's necessitated the what caused the removal of the 2 carbons?
Yeah, we'll go to it in detail, but this is the overview that I want you to see.
That's fine.
Okay, so we'll go to it in detail. Now, there's something I want you to see right now. You see, let me take this one. Okay, so now through that process, let me go back to this slide again. Okay, through that process, through that Krebs cycle process that let me go back to your no. We'll come to this one very soon. No. Okay. All right. I'm coming. Okay. All right. There's something I want to show you. Okay. No problem. I will talk about it very soon. Okay, great. There's something I want you to see here. Have you seen that the glucose that we started with? Okay, let me get one because I want you to spot something. I want you to spot something here. We started with the glucose. There's something I want you to see here. We started with the glucose. With the glucose like that. Then glucose is what? 6 carbon. 6 carbon molecule. Undergo glycolysis. Undergo glycolysis. Okay. Then give us 2 pyruvate. All right. 2 pyruvate, which are what? 2 pyruvate, each of them being what? So 3 carbon molecule, like that. Then this one undergoes, each of them undergoes decarboxylation to give us what? So the acetyl, which is what? So 2 acetyl here, which is what? 2 carbon. Two carbon. In the course of that process, let me put on top here. In the course of that process here, we've lost 2 carbon in the form of what? Carbon dioxide. From pyruvate to here, we remove, okay, because what? Decarboxylation. Okay, here. Decarboxylation. Okay, here. All right. So here we've lost carbon dioxide in the form of what? We've lost carbon in the form of what? Carbon dioxide. Then in this Krebs cycle, you realize that this 2 carbon, which is the acetyl that entered the Krebs cycle, at the end of the Krebs cycle, we've gotten, so now let's take it this way. At the end of the Krebs cycle, these 2 here, so let's say, let me put it here. Krebs cycle. Okay, at the end of the Krebs cycle, all the 2 carbon here has become what? They've all become what? The 2 carbon here Remember, so now these two acetal, so we have two of the words, so we have two here. All right, and each Krebs cycle will produce a word. Each Krebs cycle will produce 2 carbon dioxide. Okay, so at the end of the day, I have 2 into bracket. I have 2 into bracket 2 carbon dioxide, making it 4 carbon dioxide, like that. Okay, good. Now I want you to tell me something. Have you seen the glucose we started with? Now, at the end of the Krebs cycle, look at what has happened. Have you seen here? Okay. Have you seen what has happened?
Yes, Doc.
What has happened? What has happened? Yes. I want you to tell me something. What has happened? So that I will know that you understand, you've understood what I'm saying.
Please, Doc.
Yes.
Yeah, I can see that the carbon compounds, the 6 carbons,
Yes.
have now been reduced and has been sent out in the form of carbon dioxide.
Excellent. Excellent. This shouldn't have come from a man. I'm sure your wife might have told you. Yes, definitely. Excellent. So you can see that all the cis carbon, all the glucose that we've taken, when it gets into our body, by the end of the way, the second step in the cellular respiration, all the glucose, all those glucose that we've taken, the carbons in the glucose, all of them has been what? Has been oxidized into what? Into carbon dioxide. Here we are, look at it, we have 2 carbon dioxide. We have each Krebs cycle produce 2 carbon dioxide. And what? And what? And there are 2 of the process. Do you understand that? So at the end of the day, we have what? We have at the end of what? So we have two Krebs cycles. Okay, 2 Krebs cycles. Okay, all right, 2 Krebs cycles. Each of them producing what? Each of them producing 2 carbon dioxide molecules. So at the end of the day, we have what? We have ×2 of everything here. So you realize that that food that you eat, that carbon dioxide you've taken, at the end of that cycle, all those carbon dioxide, they are what? They are gone. In the form of what? They've all been converted. And all those glucose, all the glucose that you've taken, all the carbons there has been converted into what? Into carbon dioxide. Are you with me? Are you with me, my people?
Yes,
Hello?
Doc.
Yes, sir.
Are you with me? Good, beautiful. Now, so why the Krebs cycle at all? Now look at, so right now we know one of the products of Krebs cycle. We know that during Krebs cycle, one Krebs cycle produces what? 2 molecules of what? 2 molecules of CO2 gas. Okay, so and then now why Krebs cycle? Now during Krebs cycle, we have 3 molecules of NADH produced. One of what? 1 molecule of what? Of FADH2 produced. And do you know what? And 1 ATP or DTP produced. All right. So one Krebs cycle, these are the products. So one Krebs cycle, one Krebs cycle, okay, cycle. This is what we will get from it. Okay, so everything here everything here, okay, the total Krebs cycle, which is times 2, because there will be two what? There will be two of what? We have 2 of the what? Of the acetyl, okay? We have the 2 of the acetyl-CoA. So the Krebs cycle will be what? The Krebs cycle, everything here will be times what? Times 2. So times 2 Krebs cycles. Okay, cycles. So now at the end of the total Krebs cycle here, what are we going to get? We are going to get 4 molecules of what? 4 molecules of what? So remember, each Krebs cycle will produce 2. So I want the entire what? Entire Krebs cycles, because there are 2 of them. Okay, we have 2 of this produce 6 what? 6 NADH produce. We have what? We have
2
FADH produced HH2, excellent. And then we'll have
2 ATP or DTP.
excellent 2 ATP slash either ATP or GTP produced. Now, the $1 million question is this. So now look on this one on my screen, and my question is this: what is the main reason? My question is what is the main reason Krebs cycle occurs? What is the main reason Krebs cycle occurs? That's a $1 million question. Look at what I've written there.
It occurs to be
Because of what? Why? Look at the product. Look at all the products there. Look at all the products there. Look at the product and tell me. Now, before that, these 2 products, yesterday I made mention of them. I made mention of what these 2 products is. That it is what? It is something. Do you remember? I said it is what? I said something for the reduced nicotinamide adenine dinucleotide or nicotinamide adenine dinucleotide hydrogen. I said it is something. Yesterday I said it is something. Who can remember? I said it is something. Do you remember?
You said it is an electron. Something is a receiver or
electron carrier.
Carrier. Okay.
Good. Now, and this one too here, the one here too, is what? It's also an electron carrier. Okay. Good. All right. Good. So, the $1 million question is this: what is the main reason Krebs cycle occurs?
Doc, I'm trying something new.
Yes. You are scared. You are scared.
I'm not
You are
scared.
scared. The women are listening. You are scared.
I'm coming
My ladies
bravely.
are marking you. Yes, you are scared. Come, they are waiting for you.
I'm not scared. I believe strongly that
threaten you. Yes. All right, go ahead. You are listening.
I believe that paves the way for carbon dioxide to be released.
Okay.
And this one is converted from the glucose that we take in.
This is good answer, and that is not the main reason. Look at my screen. Look at what I've done. What is the main reason? What is the main reason that what is the main reason that
Krebs cycle
occurs? Look at my screen. Very well. And tell me something sweet.
Okay, Doc.
Mm-hmm.
Let's try this again.
Mm-hmm.
We can see that we have this NADH that is also produced during the cycle.
Okay.
And this one also plays an important role in glycolysis along the line. And so this Krebs cycle makes it possible for that compound to be produced so that it will be available to play its role during glycolysis.
Say something, but still that is not the reason. My ladies, where are
Sir,
you?
Sir,
Yes.
Doc, I think the Krebs cycle will generate energy for the cells which we are mentioning, like the ATP, the NADH plus the FDH.
Listen, good. You said something. I'm not doubting you. But the ATP here generated is 2, just like Krebs cycle. Only 2. Krebs cycle generate just like glycolysis. Glycolysis here, if I should go there, the topic, if I should go here, you remember that glycolysis, okay, produce what? Produced net ATP, net what? Net ATP of what? Of 2. So in the nutshell, which means that glycolysis and what? Glycolysis and Krebs cycle, they are producing almost the same amount of ATP. Yeah. You get a point, because in glycolysis you are spending 2 and you get 4. So at the end of the day, the net ATP is 2, and here the net ATP here is 2. So in a nutshell, glycolysis and Krebs cycle, there is no difference in terms of ATP production. The net ATP production, you are producing 2, I'm producing 2. All right, but I want to see. Look at what I've done on the screen. What?
Listen.
My lady, please come.
Listen.
Yes.
Let me try. So I wasn't all that active in the lesson. But
Yes.
you see four molecules of carbon dioxide up there.
Yes.
So I think it's to generate carbon dioxide.
Okay, you said something. You are right. Look at the screen again. Please look at the screen again. Okay, look at the screen again. Somebody is 4, somebody is 6, somebody is 2, and even somebody 2. They are little. Look at the screen again. Lady, tell me something. Look at the screen again. Only lady will see this. Please look at it again and tell me something sweet. Yes, my ladies. Look at it, the screen again. This one, a man will not see it. Even when I look at it, I can't see it. Only a lady will see this one. Please, where are my ladies? Yes. Okay. Look at the product. Look at the product. What you've written here? What do you have there?
All the finishing product have increased.
No. Look here. Look at the product.
Yeah. Hello, sir. Okay, so let me try this out. Looking at the screen,
Hello, sir. I've seen something.
I think one.
Yeah, say it fast. No,
you see one hydrogen.
That's what. Yes. Hey, Mr. Mark, wait for the lady to speak first. Yes, my lady, go ahead, my dear.
So let me go back and get the name right. Because I forgot the name.
Yeah, hello, sir.
The ladies are free.
Yeah, hello, sir.
Yes.
She's
free.
Yeah.
Okay, so one thing that is Krebs cycle.
Look on my screen.
These electron carriers
is the main reason Krebs cycle occurs. Main. Look at you check the molecules that have been produced. Check the molecules that have been produced.
Okay, sir. Let me try.
Yes, go ahead.
During the Krebs cycle, one cycle produced 2 molecules of carbon dioxide gas, but at the end, 4 molecules of carbon dioxide is released.
Yeah.
The same thing during one cycle. 3NADH. 3NADH is taken in, and 6.
No, they are not taking in. No. What is here is the same thing. What is here, just that the cycle is twice.
Yeah,
okay.
Twice that.
Yeah. So that's so one cycle will produce this. We produce the one here. That's why I put one cycle and put two cycles here. Okay. You are on the right track. Go ahead. Go ahead.
So during the first cycle, 3NADH. The second cycle produced twice the first one, which is the electron carrier.
Let me explain this. Let me explain this one to you. The cycle is only the cycle will be twice. Krebs cycle will be twice in the system. The reason it will be twice is that two of our acetyl-CoA is produced here. So each of them will undergo this. Okay. Not that one acetyl-CoA is going twice. No. The cycle is twice because 2 acetyl-CoAs are there. Please, you understand what I'm saying here?
Yes.
My lady, please, you understand what I'm saying?
Yes, sir.
Yeah. It is happening twice because we have 2 pyruvates giving us what? Giving us 2 acetyl-CoA. Okay, acetyl-CoA. So because 2 acetyl-CoA are there, that's why the cycle is occurring twice. So by each acetyl-CoA will go into Krebs cycle, we produce 2 carbon dioxide, 3 NADH, 1 FADH2, 1 ATP, or DTP. And so in total, okay, the cycle will occur twice in total, and that's why we have this here. And I want you to look at the product here. And remember, NADH is electron carrier, FADH is also electron carrier, and then we have 2 ATP or 2 AD, and then we have 4 carbon dioxide. And I'm asking, what is the main reason Krebs cycle occurs?
I think the pyruvate was oxidized to produce more NADH.
Okay, good. You are 80% there. So one lady should finalize it for me.
Hello, sir.
Good,
Sir, looking at I would say reactants and the products. The reactant side, we have each of them having the carbon dioxide is 2.
No.
The NADH is 2. All of them. So
no, this one is confusing. Please, let me cut you in here. What is here is not reactant. What is here? Okay, let me. I'm coming. Let me do it this way. Okay, I'm coming. Let me double the slide. Let me duplicate the slide here. Okay, and then good. I'm coming. Okay. Okay. Then let me bring this one here. So the cycle is this. This is the cycle. This cycle, what we are saying that this cycle will occur twice. That is all. And when it occurs once, this is the product. Are you with me? When this cycle occurs, this is the product. So this is the Krebs cycle. When the Krebs cycle occurs, okay, what I have here are the products. Are you there?
Yes,
But this cycle will occur twice. Why? Because we have two, this one, we have two of them present. Two of them are in the cell. So we have two of them in the cell. Okay, we have two acetyl-CoA in the cell. And because we have two of them in the cell, this cycle will occur twice. The Krebs cycle will occur twice. Are you with me? So this is the product. And because this cycle will occur twice, that is so when it occurs twice, what are we getting? Let me clean the top here. One cycle will produce this. So twice we produce that. Are you there? So not that these are the reactants. No. This is here. When this one occurs, these are the products. These are my products. 2 molecules of carbon dioxide, 3 NADH, 1 FADH, and then 1 ATP or GTP. All right. But because this cycle will occur twice, then because when it occurs twice, then if it occurs twice, then what is going to happen? When the cycle occurs twice, all right, so that is it. So here, so you can see the cycle is occurring twice, right? Can you see on my screen now? So Krebs cycle occur twice. Like that, it will occur twice. Why? Because we have two acetyl-CoA. Please, are you with me now? Krebs cycle is occurring twice because we have two acetyl-CoA. Like that. So this is, so now this cycle here is occurring twice. Why is it occurring twice? Because we have one acetyl-CoA, another acetyl-CoA here. So whatever that's happened here is times 2. That's what I'm saying. Are you there? But in one cycle, this is what you're going to get. Do you get my point? Please, are you getting it? Are you getting? My ladies, are you there? Because my ladies were missing them. My ladies, please, are you there? Are you getting it now?
Yes, sir.
Okay. Please. All right, my ladies, please. So here, each Krebs cycle, this is what is going. Each Krebs cycle will give me 2 molecules of carbon dioxide, 3 molecules of NADH, 1 FADH2, 1 ATP or GTP. Okay, so this is each cycle. And there are 2 of them because we have 2 acetyl-CoA that will be produced from the 2 pyruvate. So there are 2. So at the end of the day, at the end, I will have this. This is the total I will get. Okay. So, and my $1 million question holds, what is the main reason Krebs cycle occurs? Yes, last attempt. What is the main reason Krebs cycle occurs? Yeah, what is the main reason Krebs cycle occurs? Yes.
Please, I want to make
Hello,
my
Doc.
Valencia. No, please, nobody should talk again. Valencia said the answer long ago. Okay, so generate more electron carrier. Valencia, I was not looking here. Oh my God, I knew it.
That was what I said, dude. That was what I said.
No, I don't remember you saying it. I don't remember you saying it. No. Don't, please. Valencia, you said it around 9-1. Oh, I didn't see it. Oh, I knew only
I was saying it generates electron
No,
carriers that goes on into the electron transport system
I
or
said the
chain.
same thing, and you gave me 80%.
Oh, no,
The
no, no,
same
no, no,
thing.
no, 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 no. She said it 11 minutes ago. 9-1. That's when she said it. She said it even before I even asked the question. She said it
Please, Doc.
before
Please,
I
Doc.
asked. No, I don't want
Yes.
to hear anything anymore. The lady
I
said
said the
it
same
even
thing, and they
before.
gave me
Answer
80%.
is subject to probing.
No. She did that 1 hour ago. 1 hour ago, even before we started the class. Oh,
Dog. Dog.
so Valencia. Oh my God. I knew I was not watching the typing. Sorry, please. Valencia, forgive me. Okay.
Okay, Doc, adding on to it.
Good. All right. Valencia, thank you very much. Thank you. So you can see clearly here, we have 8 molecules of what? 8 electron carriers. Have you seen it? At the end of glycolysis, we are producing 8 electron carriers. And remember, the last step of what? Of the cellular respiration is electron transport system. Now, who is transporting the electrons there? We have the word there, NADH and FADH. And if you look at the number in terms of molecule production, the total at the end of the 2 cycles of what? At the end of 2 Krebs cycles, we are getting what? 4 molecules of carbon dioxide, 6 molecules of what? NADH, FADH, we have what? 2 of them. So in total, we have 8 molecules of what? Of electron carriers. Okay, so which means that the main reason that Krebs cycle occurs is to what? Is to make electron what? Carrier, produce the electron, the energy that has been produced, okay, has been carried in the form of what? Electrons. Okay, all right. So the main reason what? Krebs cycle, the primary carbon dioxide generation is true, ATP generation is true, but the main reason why? Because the man has come to Ghana. The man has come to Ghana, and after coming to Ghana, he is going to what? He is going to Togo. All right. Okay. So let's say Ghana is what? Ghana is a Krebs cycle. Okay. You've entered the Krebs cycle. Now you are going to Togo. Now you are moving from Ghana to Togo. So let's say Togo, we don't accept what? Cities there. So this man needs to change the word, the cities into what? Into safer. So now the electron carriers here, they are the safest are the what? The NADH and FADH. So the main reason Krebs cycle occurs is to generate the electron carriers. Thank you very much. What an answer. Okay. Thank you very much. Okay, all right. So now we know the Krebs cycle. Here, I don't really, so these are the, let's see. These are the products of what? The Krebs cycle. All right. So look at the products of Krebs cycle here. So Krebs cycle, these are the detailed stuff here. So we have the Krebs cycle here. This is the Krebs cycle. That's it. We will enter into the citrate, react with the what? Oxaloacetate to produce the what? To produce the citrate. And then 1 FADH will be produced, 3 NADH will be produced, 1 ATP or GTP produced, and then 2 carbon dioxide will be released. At the end of the day, we will generate our oxaloacetate back. Okay, so the primary reason of what? Of Krebs cycle is to what? Generate what? Generate that chemical energy, that chemical potential energy in the form of what? In the form of NADH. Okay, because at the end of the day, we are having these two electron what? Carriers. So the energy that has been ATP will produce, all right, but remember, just 2 ATP will produce, just ATP. But the vast majority of what? ATP produced will be in the form of what? NADH. All right, that will be taken to the what? To the electron transport system. Are you there? So now the energy there is in the form of electron. So these ones are carrying electrons, high energy electrons. Okay, so they are carrying high energy electrons to the what? To the electron transport chain. So the main reason Krebs cycle, okay, the primary reason is to generate what? Energy in the form of what? In the form of what? In the form of or it generates the electron carriers. Okay, so you see that we see something like a lot of what? Potential energy. So this is more or less a chemical potential energy. All right, okay, in the form of what? The electron carriers. Six this to that. Okay, all right, so that is the primary reason. Okay, now let me take you through a little bit of what detail. Please, I don't expect you to put the whole of this cycle in your head, okay, but I want to just catch on some salient points that I expect you to put that one in your head. Okay, so so you can see the cycle. So the key ones that I want to touch on is this one. So you can see the oxaloacetate, here. The acetyl-CoA will enter in the form of the acetyl will enter there like that, and then when it enters there like that, now we have an enzyme called what? We have an enzyme called citrate synthase. So the most of the time, anytime you see the word synthase, anytime you see the word synthase, it means that the enzyme is producing something. It's usually producing what is coming in front of it. So if I say citrate synthase, the moment you see that, it means that the enzyme is facilitating this production like that. Okay, so if I say ATP synthase, it means that this enzyme is producing what? ATP. All right, okay, this enzyme is producing ATP. Okay, good. Now, so back to so here, if you look at this, you can see, so that acetyl-CoA will enter here. All right, we get the enzyme that is going to catalyze the process is the what? Is the citrate what? Synthase. Okay, the citrate synthase, and then we'll form our citrate. Okay, then it will undergo a little bit of what? A little bit of isomerization. Okay, we get something like what? Citrate, and then the enzyme there is called the what? That's the aconitase. Okay, please, the enzyme that is important to me for you to put in your head is this one. Okay, this first two reaction, this is the one that is very, very important to me, that you should put it in your head. But the reason I'm going through the cycle with you is so that I want you to see where the NADH is produced. Okay, but I don't really expect you to put the entire cycle. The most important thing I want to put in your head is this, is what is here. Okay, this is what is here is the most important thing I want you to put in your head. Okay, all right, good. But I want you to appreciate how the NADH was came about. Okay, so we have this one. So after the production, there's some sort of a hydration work. Water will be added here. We have the aconitase, and then we get our what? Our citrate. Then so it is on the what? So this is the first step. Second, on the third step here, this isocitrate, okay, will be converted into what? Into alpha ketoglutarate. Okay, into alpha ketoglutarate. And during that process, okay, we have what? The NADH will be converted into what? Into the NAD class will be converted into NADH. Now, remember, look at the enzyme here. Have you seen the enzyme here? Anytime hydrogen is removed, okay, anytime hydrogen is removed, you see the enzyme there is called dehydrogenase. Are you there? Anytime we remove, so anytime hydrogen is removed, you see, when the hydrogen is removed, somebody should be there to pick the hydrogen. And so when hydrogen is removed, you have the NADH should come to pick it up. Are you there? Okay, should come and what? Pick it up. All right. So you can see in this process from the isocitrate to alpha ketoglutarate. All right, we can see that one NADH has been produced. Then from the what? From the alpha ketoglutarate to what? To the succinyl coenzyme A. Okay, you can see that during that process, another carbon dioxide has been taken out. So you can see that the carbon dioxide production here, it is just from where? Just few steps away from the what? From the citrate. Just few steps away from the isocitrate to the what? To the succinyl coenzyme A. Just that space. Okay, just that space, all the carbon dioxide will be taken out. Okay, so you can see here so remember this one is still what? It is still, here is 6 carbon molecule. Here is still 6 carbon molecule. But as carbon dioxide decarboxylation has occurred here, so you can see that alpha ketoglutarate is how many carbon molecules now? Yes, it's how many carbon molecule? Yes, it's how many carbon molecule? The alpha ketoglutarate is how many carbon molecule? Yes, it's how many carbon molecule, please? My lady, are you there? Yeah, how many carbon molecule, please?
5.
5. Excellent.
5.
Excellent. So alpha ketoglutarate is what? It's a 5 carbon molecule. Why? Because from the isocitrate, okay, one decarboxylation has occurred. One carbon has been lost in the form of what? Carbon dioxide. Okay, and then between the alpha what? Ketoglutarate to the what? To the succinyl what? To the succinyl coenzyme A. Another what? Another decarboxylation, as well as the what? Conversion of what? NADH plus to NAD, conversion of NAD plus to NADH, okay, will occur there. So just from isocitrate to what? To succinyl coenzyme A, the 2 carbon dioxide molecules, okay, because from the what? From the isocitrate, we go to the what? That's the alpha ketoglutarate, and from alpha ketoglutarate, we go to the what? That's the succinyl coenzyme A. So just that 2 steps of reaction, the carbon dioxide are all gone. And so you can see that the succinyl CoA from here all coming, all the carbons there are all 4 carbons. All the carbons there are all 4 carbons. Okay, you can see from here coming. The carbons coming here are all what? 4 carbons. Okay, so from here, so from isocitrate to succinyl CoA, we have what? 2 NADH has been what? 2 NADH molecules have been produced. And in between what? In between the succinyl CoA to succinate, okay, it will result in the what? In the breaking down of what? The breaking down of conversion of what? Of GDP to what? To GTP. All right, and this this GTP can be converted into ATP. All right, that is why we always say that we always write GTP stroke what? ATP. So from the what? From the succinyl CoA to the what? To the what? To the succinate. All right. We have our GTP produced, but this GTP can undergo reaction to form what? ATP. All right. Then between our what? Our succinate. Okay, between our succinate to fumarate. Okay, from succinate to fumarate, we have what? Our FADH2 is produced. All right. Okay, and then the last but one step is the malate. Okay, from malate to the production of the what? The oxaloacetate. Okay, so you can see right from this side after the alpha ketoglutarate. Okay, all the molecules we are getting here. Hey, our mothers, please. Okay, we'll go for a break right now. 2 minutes time, please. We are breaking for. We'll go for a break. Okay. All right. So from here going, you see from the alpha ketoglutarate, you can see all the molecules here. They are all 4 carbon molecules going. All of them are 4 carbon molecules to generate what? So the next place where our NADH was produced is the what? Is the malate to the what? To the oxaloacetate. Okay, that is where. But you can see that in all the reaction, even though this reaction is occurring in presence of oxygen, you can see that the oxidation that is occurring here is more of removal of hydrogen. Please, have you noticed that? Have you seen that? Have you seen that even though oxygen, we require oxygen for this reaction to occur, the oxidation that is occurring here is more of removal of what? Of oxygen rather than what? Rather than addition of removal of hydrogen rather than addition of what? Oxygen. I don't know whether you've noticed that. Please, have you noticed that? You can see all of them. You see dehydrogenase. Okay, so isocitrate dehydrogenase, alpha glutarate dehydrogenase, succinyl synthase. This one is going to produce the what? The succinate. From here, succinate dehydrogenase, malate dehydrogenase. So all of them, you can see removal of what? They remove the hydrogen, and then when they remove the hydrogen, you can see somebody should be there to pick it up. Are you there? Okay, so you can see in the reaction, in this reaction, how many oxidation steps are there? How many oxidation steps are in the Krebs cycle? How many oxidation steps are in the Krebs cycle?
There are 4.
A gentleman said it. That's good. Good. Okay, I thought a lady said it. Please allow my ladies to speak first. Okay. All right. Good. Excellent. Well done. Well done. So in Krebs cycle, there are 4 oxidation reactions. Okay. And how do you get the 4? Please, you don't need to go through all this cycle to know that there will be 4. The quick way to know that is here. The quick way to know that. The quick way to know that is here. Is here. Because in this reaction, in the first reaction here, remember 3, 8, this one means 3 oxidation, okay, all right, and then this one is what? 1 oxidation. Are you with me? 3 oxidation and what? And 1 oxidation. All right, here. All right, so in total, 4 oxidations are occurring in the Krebs cycle. And how many decarboxylation reactions? How many decarboxylation reactions occurs in a Krebs cycle? How many decarboxylation reaction occurs in a Krebs cycle?
2.
Excellent, 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 excellent. Okay, so in a Krebs cycle, in one Krebs cycle, we we have we have 4 oxidations. Okay. 4 oxidations. All right. And what? And 2 decarboxylation. All right. 2 decarboxylation. All right. Okay. Carboxylation. All right. Good. All right. So that the 4 oxidations, they are generating 3 ATP and what? And 1 Fa, 3 NADH and 1 FADH2. All right, as electron carriers. Okay, please let me give you 30 minutes break. All right, is that okay? Or we should kill it. Okay, let's give ourselves some 30-minute break. Oh, Ademan,
Yes, sir.
please let's give ourselves a little bit of a break. Okay, because all right. Okay, so when you come back from break, all right. So here, the same thing that I have put in a simple form so that you know. Okay, 5 carbon 5. So you can see that this one entered, away, disappeared. 6 carbon here. Move on to 5 carbon, and from 5 carbon, everything move all the 4 carbon, 4 carbon, 4 carbon, all the way here. So this is more of the summary of it. Okay. All right. Okay.
Doc.
Hello. Yes. Please go ahead.
Please kindly put the last slide, the one you wrote on. I want to make some notes from it.
Okay, the last one.
Yeah.
This one. Oh, no, not this one.
I think it's a Krebs cycle.
This one, the Krebs cycle. No, not this one. The cycle here. Okay. Okay.
So, no, the one where you wrote the 4 oxidation.
Okay.
And then
was it on a new slide? A new? Yeah. Where is it? Where have I put it? Okay, let me write it again for you. Let me do it for you. Okay, so in a way,
sir, it's over there. The one that you flip over, it's over there. Just go back. Let me show you that one.
This one?
I just joined. Yeah, this one.
This one.
I think
This one.
Oh, okay,
Yeah,
okay.
this one.
So one Krebs cycle will give, in one Krebs cycle, a reaction 4 oxidation, all right, and what? 2 decarboxylation, all right, good. And remember, our oxidation here is more of what? Removal of what? Of hydrogen, okay, all right, and then the what? The decarboxylation is removal of what? Of carbon dioxide. Okay. All right. Okay. So this is one Krebs cycle. That's what you are going to get. Okay. Right. So when you come back, then okay. So okay, you are making the note. You're taking. All right. Ten o'clock on the dot, please. We are in 10 o'clock on the dot. Okay. All right. Okay. So in case you want to make notes on this one as well, okay, let me put it here so that at least you can also appreciate what happens. Citrate, isocitrate, okay, alpha glutarate, okay, succinate coenzyme A, succinate. From succinate to what? Fumarate, fumarate to malate, malate to okay, malate to the oxalate.
sir, are you going for break or you are still teaching?
Oh no, I'm just mentioning what it's. I'm not teaching. The power.
Okay, so let's go for the break.
So let's go for the break. Okay, so please 9 o'clock on the dot here.
Doc, please kindly leave your diagram.
The previous
On
one.
the screen
The
for
one before this.
your own diagram.
But you say you are also going for break.
We are writing something down, please.
Definitely, when we come back, you revise before we go. Moreover, you will send the slides to go for the break and come.
Okay.
Wow.
No. Nó mình mà bữa cái này là cái gì đâu. Cái gì đâu thì nó. Đâu có anh cái gì chơi cả. Anh mà anh mà
faced with personal experience, students enjoy peer relationships and collaborative learning, using multiple problem solving, then safe learning environment.
All right, Nico, I can see your hand is up. Yes. Okay. Nico, I can see your hand is up. Is it down? Okay. Please try your hands on the true or false that I have on the screen. All right, please try your hands on the true or false on the screen.
Doc, this one there, I scored 10/10. But since you don't want to hear male voice, I'm on strike.
No, you finish. Everybody finish, and then I'm just giving you just 3 minutes, and then
Hello, sir.
hello.
I believe it will be part of the slide notes.
Yeah, so like we'll just do it quickly. 1 minute more, then we'll do it.
Okay. I think I'm done. Only why plenty force?
Hey, come on for there. Who you tell you say plenty force?
Plenty force, sir.
See this battle.
Please don't do that.
Hey,
Don't do that.
hey,
We are
Massa,
done and we want to. No, don't do that. Don't do that. It's wrong.
Massa,
It's wrong. Don't do that. Don't do that.
Massa, keep quiet. If you are done with it, just keep quiet.
Yeah,
Don't want any trouble.
Please,
Don't come and
relax,
deceive
so that
my
we finish.
ladies here. Wait.
Popo,
Don't
please
deceive my
relax.
ladies here.
Let's
Massa,
finish.
Unless we
Massa,
said our
don't come and
ladies, some of
deceive
the
my
others.
ladies here. I don't like that. Don't come and
Take
deceive
your
my
time.
ladies.
Yes, take your time.
Yeah. Fidele, thank you. Okay.
Yes, sir.
As for this Fidelia, I will mute you will not come to this platform again. This is the last time.
The man relax. Oh, why? The man relax. Let's finish.
Fidelia, don't mind him. I would
Yes,
rather mute him.
Thank you, sir.
When you finish, go to him for your slides and you call me again, you see what I'll do to you.
Sorry, Mr. Popo, sorry.
You are no romantic. I'm disappointed.
When it comes to academic, no romance. So.
See your life.
Because
all right,
if you go to the job market,
1 minute more. 1 minute more.
In the job market, girls always have the phone.
Hello. Yes, please, Bashir. 1 minute more, and then we can.
Please, Doc.
Yes, please, I'm listening.
Please, Doc. Hello.
Hello, please, I can hear you. I'm listening.
Yeah, please, Doc. Yeah, please. Are you free? Please, are you hearing the screen? I can't see you. Are you?
You can't see the screen.
Yes, I'm saying that. Yeah.
You can't see the screen.
Yeah, I used to say why I cannot see it.
No, the screen is on. There is
yeah.
Bashiru, exit and come again. Exit and rejoin.
When I go to please, I go to
So
number
exit,
8
exit
and
and
I
rejoin. Exit and rejoin.
So exit and come back. That is all.
Sure.
Yeah, you see how eco-frantic you are. When I get the answer, you say, wait, quickly come and put your answers down there.
Please don't
But
put
we
your
are
answers.
putting the
Please.
answers. Or
No, don't post your answer, please. Don't post your answer.
I didn't know. I thought
Please
you
don't
were
post your
supposed to
answer.
post.
No, don't post it.
When I say it was made up
Alright,
of
it's good
two
your answers
force,
are here
so
though.
many force. We say, wait. You can't put your own two there.
Hey, Adiba, why this attack? By you, what's up?
That's when I go breaking leg, making no leave out.
I thought that to post, but not to say. Sorry.
Okay, stop work. Okay, stop work. So let's do it together. Number 1, conversion of pyruvate to acetyl-CoA is catalyzed by pyruvate dehydrogenase. So false.
False.
Glycolysis produces net ATP of 4. That's
it's false.
And yes, in the formation of acetyl-CoA, the formation of acetyl-CoA produces 2 ATP, 2 ANDP in total. That is false. During the formation, that's false. During the formation of acetyl-CoA, no ATP is formed. The intermediate process doesn't have ATP. It produces that, but no ATP.
Yeah.
You have 2 truths. The rest are
dead. There are 3 oxidation reactions in each Krebs cycle. That is false. It's 4.
Correct. I've passed.
And then for a molecule of glucose, the Krebs cycle generates 2 carbon dioxide, 3 DNTP, and that no. For a molecule of glucose, everything here will be doubled. So for a molecule of glucose, carbon dioxide, so it is false. Everything here is doubled. Right. So here for a molecule of glucose, remember the pyruvate will be 2. And so the Krebs cycle, they will generate 2 acetyl-CoA. So the Krebs cycle will be here times 2. Here times 2. So it is 4. So for a molecule of glucose, 4 carbon dioxide, 6 NADH, 2 ATP or GTP, and then 2 FeCl2 produced. The first step in Krebs cycle is the reaction between oxaloacetate to form isocitrate. That is false.
False.
Okay, it should be right. It's to form citrate. Okay. Unlike acetyl-CoA formation, Krebs cycle occurs in the mitochondria. This is also false. Acetyl-CoA formation also occurs in the mitochondria. So the unlike here with this one, acetyl-CoA also occurs in the mitochondria. So the answer, this one is also false. That's a very tricky question. Okay, so it's false. Let me take this one out here. Let me take so that I can put it there. Okay, let me see. Okay, let me reduce the size here. Let me make this one 29. Okay, so that I can put false here. Krebs cycle, okay, it's only in the presence of oxygen.
False.
That is true.
It's true.
Ajiman, you are dead. Ajiman, you've got about 3 wrongs. So. Me, I've
Oh, no, I wasn't part. I was not part
Ajiman.
of this lesson. No, I'm just using my mind.
You
Went
did
past.
it.
I didn't get
Ajiman.
anything.
Don't want a
Ajiman.
situation you finish and come and tell us you had
I
10
know I've
over 10.
gotten 2 wrongs. I know I've gotten 2 wrongs. So I'm not shy.
Conversion of isocitrate to ketoglutarate is a carboxylation process. That is true.
That is true. Good.
And then carbon dioxide, NADH, and FADH are all electron carriers. That is
false.
Carbon dioxide is not an electron carrier. Okay. All right. So who had 10 over 10?
No,
I had 10 over 10.
It's a lie.
Oh, okay. Let's see Prosper. Prosper had, let's see, 4. The first 4 are all false. Number 4 is false. So Prosper, okay, no, that's the Nyako. So Nyako, you missed the 4. Number 6 is false. Number 7 is false. 0 is true. So you missed that one too. Number 8 is true. So you missed 3. You missed 3. Yaku, you missed 3. You missed 3. You missed 3. And then Prosper, number 4 is false. You missed that one. And then number 6 is false. You had it. Number 7 is false. You missed this one too. You missed 4. You missed 7. And then 8. You had that one. Okay, so you missed 2. 8. Oh, Belinda had 10 over 10. Oh, that's great. Oh, Belinda, I trust you. Well done.
AI.
What is AI? Then look at Alan, a 9 over 10. 9 over 10. The man 7, 8. Yeah. No has written. Oh, Savannah, well done. Savannah. Yes. Oh, okay. Owusu 8. Johnny 6. Ajiman, how did you get the 8 over 10? Well, Charlie, I said I can't catch.
I said it. I said it. I had 2 wrong.
We catch 8 7. You get 3 of them.
No.
What
that is what I said. You are not out of my marks. I know my
Hafiz Muhammad. Hey, Hafiz
Muhammad.
Muhammad.
I was not part of this lesson.
King
the
Hafiz.
that I didn't join.
Yeah, Alexi 7, not bad. King Hafiz 8. Lord the Kingsley 9. Well done. Oh, Juliet 7, not bad. Alex 7. Paul 8. Good. That's good. Not bad at all. That's good. Okay, that's quite impressive. Well done. Okay, that's very good. Okay, I think well done. You've done well. Okay, good. So enough of what? Enough of Krebs cycle. I think you've done very, very well. Okay, so I'll keep this slide surface here for you. Okay, I'll keep it for you. Okay, so that okay. So the next I'm going to talk about is electron transport system. Okay, so electron transport system is not really difficult at all. Even I doubt today we want to finish because they are going to write quiz for me. So at exactly 11 o'clock or 11:05, we need to pause the class. So what we will do is that what will be left with only photosynthesis, which I could do that one in 1 hour. So that one, Ajiman will find time so that we'll do it. But I'm so happy you fully understood this one very, very well because this one at SS level and your master's level and stuff like that, you need a lot of that. Okay, all right. Good. So we're going to talk about electron transport. But before you go to talk about electron transport, let me give you the overview here before we go to the slide itself. Okay. Now, electron transport system. Now here we call something that system, the electron transport system, collectively we call it what? We call it oxidative phosphorylation. That is it, okay? Oxidative phosphorylation. Okay. Now, let's see what does it mean. Oxidative phosphorylation, there are 2 processes there. What are they? The 2 processes there are electron electron transport system. We always mention electron transport system, but this is advanced level. So let me explain both to you. System, and another thing we call it chemiosmosis. Chemiosmosis, like that. So these are the two main processes that tend to occur. These are two main processes that tend to occur. Okay, we have the chemiosmosis and electron transport system. Now, electron transport system, what is their love language? When you go there, what electron does currency electron transport system accept? It's NADH and NAFADH2. So all those ones that have been generated, this is what they are going to do. So they are here. Okay, these are the what? These are the two electron carriers. So these two electron carriers, they have brought their energy in the form of what? Electron. So okay, they have brought their energy in the form of what? In the form of what? Electron. Okay, and these, so let me write this one. NADH carries more energy than FADH2. All right, don't forget that they are all electron carriers. One carries more energy, more electron energy than the other. So FADH carries what? More energy, more energy than what? Than FADH2. All right, and electron transport system. Okay, this system, this oxidative phosphorylation, these two systems, they are actually occurring in something we call, they occur in the what? Intermembrane. Okay, the intermembrane. So the inner membrane, the intermembrane. Okay, so these two processes, they are occurring in the what? The intermembrane. Okay, the intermembrane space, as well as intermembrane stroke what? Space, the membrane, the intermembrane itself, as well as what? Intermembrane space. Intermembrane space. All right, good. So here they are actually associated, the oxidative phosphorylation, they are associated with what? Intermembrane, what? Membrane space and the intermembrane. Okay, all right, good. So let's see what happens. So now let me go to the diagram. Okay, of the what? Of the mitochondria. Okay, beautiful. Now let's see the diagram. Now let's go to our diagram. Now you see, when you come to this diagram, this is where we have the metrics. Okay, so well, maybe I can sketch some mitochondria quickly here. Let me draw mitochondria quickly here. Let me draw mitochondria. Okay, let me see what I have to do, some drawing here. So now let's say this is my mitochondria, like that. So it has this sausage-like thing, like that. Okay, then another membrane there, like that. Another membrane there, like that. Okay, good. So this side here is what we are referring to as what? As intermembrane space. Okay, so there is what? There is we have what? We have outer membrane. Okay, we have outer membrane. Okay, like this. Okay, we have the outer membrane like that. It's double membrane bound, outer membrane like this. And then we have inner membrane. Okay, so that's the inner membrane. Now, so when we talk of what? So this is the outer membrane here, and this is my inner membrane. And what is here is intermembrane space, this side. Okay, between the two membranes. Okay, so inner membrane, this is inner membrane here, and this one is the what? The outer membrane. outer membrane here. And what is here? Okay, let me pick different color. So what is here? What is here is the word. What is here inside here? That's the intermembrane space. Okay, so that's the space between the two membranes. All right, of the organelle. And this side here is the matrix. This side here is the matrix. Okay, so the C here is the matrix. So this is where the acetyl-CoA formation and the Krebs cycle occurs. Okay, so Krebs cycle occurs in this space. So this is the matrix, like that. Okay, so at the end of the matrix, what is happening now? So what you have here, so you can see that this side is the matrix, mitochondrial matrix, and this is the membrane. This is the membrane. This is the inner membrane, and this side here is intermembrane space. So this side here is my membrane space. So outer membrane here. This is my membrane here. Okay, so space between the two membranes. So that is the intermembrane space. Okay, intermembrane space. All right, intermembrane space like that. Okay, great. So what is going to happen? Now, what is going to happen is this. Okay, we have, so this is the intermembrane space here, and this is the mitochondrial matrix. Okay, intermembrane space is here, and what do we have here? These are the all these structures are protein complexes. They are protein complexes in the intermembrane space. Okay, so these are the protein complexes in the intermembrane this intermembrane space. These are protein complexes in the intermembrane. This is the membrane. This is inner membrane. What is here? All these sides are all inner membrane. This is the intermembrane space. This is the matrix. So what is going to happen? Now, what is going to happen is that the electron carriers will come here and give their electrons. So let's see what happens. Now you can see that NADH has come. It is going to release its electron, and when it will dissociate. When it breaks, when it dissociates, so you see now NADH is reduced. Now it's going to be oxidized. When you say you are going to be oxidized, it means that they are going to what? You are going to remove the hydrogen ions you have from you. Now, when hydrogen ion is removed, okay, the electron generated will be passed on. So you can see that after you remove this one, okay, the electron generator, remember, as I told you yesterday, let me go back to my slide here. Now, when something, let's say it's undergoing oxidation like this, okay, oxidation like that, okay, so it can become like this. So Cu2 plus ion plus what? Plus 2 electrons. Plus 2 electrons like this. So this one, so when it get there, so here we can say that what it says is undergoing oxidation. What is here is undergoing what? Oxidation. All right, oxidation. So you can see this one is being what? Oxidized. So when it's oxidized here, oxidation can be defined as what? As removal of electron. So you can see that, okay, so these are the protein complexes in the inner membrane. Okay, we have complex 1, complex 2, complex 3, complex 4. What is here is called electron. This one is a mobile carrier. Okay, they are complex there, but they are mobile. Okay, so ubiquinone, and we have another one, we call it this one, like this one, the cytochrome C. This one, and this one, they are mobile electron carriers. Okay, we have complex 1, complex 2, complex 3, complex 4. So let's see what is going to happen. So what is going to happen is this. Okay, when they come here, everybody is coming to do what? Everybody is coming to donate, come and give their electrons. So when you come, this one will undergo oxidation. Okay, this one will undergo oxidation like that. Okay, this side. So let me put arrow here. What is here? It doesn't really seem correct. Okay, this one is undergoing oxidation. Have you seen it? It's undergoing oxidation like that. And when it undergoes oxidation, what does it do? You can see, look at the two products it's producing. Okay, so it undergoes oxidation. All right, if undergoes oxidation, okay, like that. Okay, like that. So when we undergo oxidation, we are going to get this hydrogen ion and NAD plus ion. Okay, we'll get those hydrogen here in NAD plus. So now look at what is going to happen. So this one, this will be released. This hydrogen ions here, removal of hydrogen ion. Then this one, this hydrogen ions here. Okay, look at that. So the electron that will be given out as a result of oxidation will now be passed on from one chain, one protein complex to another. So you can see that this one has released its electron. So here the NADH could give its electron to a complex 1, another one, and then another one, FADA will come and give it to complex 2, NADH again will give it to complex 3, and the electrons are being passed on. Now the energy in the electrons will be used to pump these hydrogen ions to the intermembrane space. Are you with me? Let me take it one more time. What happens is that we call the electron transport chain. Now, so as NADH and FADH, when they are going, they are containing what? They are containing high energy electrons. So these high energy electrons, they are going to do what? They are going to transport this high energy electrons here, okay, to the electron transport system in the what? In the electron transport system. When they go there, this NADH will break down, releasing hydrogen ions and FAD. Okay. Now, this hydrogen ions, the electrons will be passed here. Look at that, the electron will be passed on like that, like that, like that, like that. As they've been passed on, the energy in it is used to pump these hydrogen ions into the intermembrane space. So the energy that is given, remember there are more hydrogen ions in the intermembrane space than hydrogen ions here. So if we are going to send hydrogen ion to this side, then it means that you are supposed to use what? Energy. So that's what we call it pump. Okay. Now this energy is coming as a result of what? Electrochemical gradient. Okay. It's not, this energy here is not using ATP. It's only using the energy from the passing of what? The electrons. Okay. The energy released from the electrons as they move from one protein carrier to another protein carrier to another protein carrier. All right. As they do that, okay, so they release, so this one, FADH, will release its what? It's hydrogens, okay, its electron. And when it releases its electron, the energy that will be released from the what? From the electron will be used to what? To pump these hydrogen ions into the what? Intermembrane space. All right. And so as they passed on, okay, so here, FADH gives its electron to what? To the complex 2. And then the rest, the NADH, okay, will give it to what? 1, 2, and 1, 3, and 4. Okay. So they will use the energy here. They will use the what? That energy from the electron to pump the hydrogen ions to the intermembrane space. Good. Now, after the hydrogen ion pop into the intermembrane space, what is going to happen? And then finally, oxygen is there to catch this electron. Okay. After going through all the carriers, then we have what? Oxygen here, and then the oxygen will catch, okay, the electron, and then in conjunction with the hydrogen ion, form what? Water. Okay. So the electron here is going to reduce oxygen into what? Into water. All right. Remember, addition of electron or addition of hydrogen is what? Is reduction. So when this electron is passed, there should be oxygen there to catch it. So it will catch the electron here, and then with presence of the hydrogen ion, you form what? Water. Okay, that's very important process. So you know, at the end of respiration, you know where the water is coming from, because the electron will be used to what? To reduce the oxygen in presence of hydrogen, to form what? Water. Now, look at what is going to happen. Now, after pumping all these hydrogen ions up here, what is going to happen is that you can see we have high concentration of hydrogen ions here. Now, have you seen this big thing? And this big thing here is called what? It's called so it goes like this. We have something like this. So protein carriers are coming. NADH and FADH2. All right. So all these ones are protein complexes with some carriers here, mobile electron carrier, protein complexes. There are 4 of them here like this. Okay, with another carrier somewhere here. Let me take different color. All right. So this is a mobile. But maybe let me use red. Red. Okay, red here. Okay. So this is the mobile carriers. Okay, mobile carriers. So these are they are here. So when this one reaches here, it will pass its electrons. It will release its electrons like this. An electron will be passed. Will be passed. Passed. So the energy from the electron, this electron transfer, will be used to pump hydrogen ions here. There are plenty of hydrogen ions here than this is intermembrane space. So here outside there is what? Intermembrane space. And this side is what? Matrix. This side there is the matrix. All right. So you pass on like that, you pass on like that. Now, and then at the end, there should be oxygen here, O2 here to catch this girl. Like that. So oxygen here to catch this girl, and in presence of what? Hydrogen, in presence of hydrogen, okay, we end up forming what? Water molecules. All right. So as electron is being passed on from one complex to the other, this 4 protein complexes here, and then with 2 mobile, okay, mobile electron transporter. Okay, so these are the carriers who have come to release their oxygen, their electron. So as they pass, the energy from the electron is used to pump hydrogen ions here, hydrogen ions, plenty, and hydrogen ions there, hydrogen ions there, hydrogen ions there. Now there's something here, we call it what? Here at the end here, there's this big thing here. So let me make it something like a turbine. Okay. Now, so this hydrogen ions here, after everything, these hydrogen ions will now begin to flow down here. So the flowing of the hydrogen ions through this structure. Okay, let me use different color for it. Okay, let me use different color here. I want a bigger. So the flowing of what? The flowing of the hydrogen ions through this what? This structure. This structure is called what? It's called ATP synthase. Okay, ATP what? Synthase. Okay. It's called what? ATP synthase. All right, so this particular molecule, and do you know what it does? Now, as the hydrogen are flowing here, the energy in it, the energy in this one, okay, the energy in this one is used to convert ADP, okay, plus what? They will add inorganic phosphate to it and convert it into what? Into ATP. Now, the flow of the what? Of the hydrogen ions through the what? Through the synthase. So the synthase acts like what? It acts like the turbine. Like you see, if you go to Akosombo, all right, if you go to Akosombo, so the turbines are rotating, and as the turbines are rotating, what happens? As the turbines are rotating, then the what? They are using it to generate electricity. So the equivalent of the turbine in the what? In the intermembrane, the inner membrane is the what? Is the ATP synthase, which is this one, which is this green big thing here. So they will allow this hydrogen ion to flow through. And the flow of the hydrogen ion is called a what? It's called, this is the flow of the hydrogen ion is what we call as what? What is called chemiosmosis. Okay, so flow of the what? Of the hydrogen, flow of the H plus ions through the what? Through the ATP synthase is what we call what? Chemosmosis. Chemosmosis, okay? All right, chemosmosis, like that, okay? So that the flow of what? That flow is what we call chemosmosis, okay? And during that flow, what happens? As this hydrogen ion goes through the what? The turbine here, which is the what? The ATP synthase, then ADP, inorganic phosphate is added to ADP, that's adenosine dinucleotide phosphate, to get what? ATP. So the vast majority of the energy produced is coming from here. The vast majority of the, so here, okay, the vast majority of the energy produced from what? Cellular respiration is coming from here. But let me tell you what exactly is happening here. Now, what is happening here is this. It's very simple. Now, you see, when the NADH, when they bring this thing, the NADH is carrying equivalent of what? One NADH gives equivalent of 2.5 ATP. Okay, one ADH, after releasing the electron, the amount of energy it produces is 2.5 ATP. And then the FADH2 gives energy equivalent of what? 1.5 ATP. Are you there? Good. So do you know that we can calculate the total number of what? The total number of NADH, and then we can calculate how much energy is produced. Okay, so do you know there's this nice question I normally give? Okay, I normally give, all right? Now, from here, how much, using this, how much ATP is produced, okay, from, okay, how much energy, let me put it how much energy, using this, using what is here, using what is here, using this one, using this, okay, how much ATP is, oh no, how much energy is produced from the Krebs cycle. I want to see who will get this one. Remember, in Krebs cycle, we have total produces what? 6 NADH, 2 FADH2, and then 2 ATP. All right. So if we do this conversion here, how much energy is produced? Remember, energy, we are calculating it in terms of what? In terms of ATP. How much energy is produced in terms of ATP? From Krebs cycle alone.
So, sir, you clean the first part of the question.
How much?
Okay,
Thank
you.
How much ATP? Okay. How much? Oh no, I didn't do it well. How much? Oh no, I repeated the question. So how much? Good. How much energy is produced from the Krebs cycle in terms of ATP?
Thank you.
All right, who has finished? How much energy is produced from the Krebs cycle in terms of ATP? Who has finished? So if I had to calculate, it will be here 6 times 2.5. Okay, and that will give us what? That will give us what? 15. Good. In here, we give us 2 times 1.5. And that will give us what? That will give us 3. And then the normal ATP that will be produced is 2. So in total, you are getting what? You are getting 20. So to total to give us what? 15 plus 3 plus 2, 20 ATP. Have you seen it? So if we are to total all the ATPs in cellular respiration, let's see what we are going to get. Let's see if we are going to total all the ATPs in cellular respiration, what we are going to get. Glycolysis. In glycolysis, we had what? We had net ATP of 2. Net, we only use the net, okay? Then we had what? We had okay, let me go to the next slide. Okay, so let's see. In glycolysis, net ATP of 2, but we have what? We had plus what? 2 NADH. And this 2 NADH means that in glycolysis, what are we getting in total? That NADH produced here, NADH will give us what? 2 times this will give us 5. So I will get 2 plus 5. Glycolysis, that's what we are getting. And then acetyl-CoA formation, acetyl-CoA formation gave us what? Give us 2 NADH. That is also 5. That's equivalent to 5 what? So 2 ATP here, ATP here, and then 5 ATP there. ATP. Then this acetyl-CoA formation in the intermediate will produce what? We produce 2 NADH for each of them. So you are going to get what? You are going to get 2 times 2.5 will give you 5 ATP. Then finally, the Krebs cycle, okay, Krebs cycle, okay, in conjunction with what? Electron transport, okay. Okay, so let's see, the Krebs cycle gave what? Gave total of what? The total of 6 NADH 2 FA DH2 and then 2 ATP. So in total, we know that this one in total we just calculated it up there. So when we get here, all the conversion here of this one, this side, this at the transfer at the T electron transport chain, transport chain, all the NADH and this will be converted. So after that one, after the conversion, we'll get here total we are going to get. I think we've calculated it up there, which gave us what? 20 ATP. Everything is 20 here. So let's see. So now when you convert, so here will give us 7 in the ATP here. Here give us 5 ATP. Okay, 7 here, 5 here, and 20 here. So in total, we are getting what? 32 ATPs. From what? From complete what? Complete cellular respiration comprising aerobic, anaerobic, and aerobic respiration. All right. Please, do you understand the process? Remember, let me ask you this question. If there is no oxygen, how much net ATP will be produced?
Two.
Well done. That's a very tricky question. Why did you say 2?
7.
Why did you say 2? Why did you say 2? Because this one is here. Why did you say 2? This one is here, right? So why did you say 2? Yes. Why did you say 2? Because this at this phase here is not energy yet. It is when it gets to the electron transport chain that this one could be converted into energy. But when it is here, it is not energy yet. So here, at the end of the Krebs, at the end of the glycolysis, the net ATP is just 2 here, and this is produced. But it is this one when they take it from here to the what? When they take this one, okay, and this one's here, this one here, when they take them to what? The electron transport chain, that's when they will convert them into ATP. But until they get there, it will only be this. So without oxygen, only glycolysis will occur. it will produce this all right, but this one will not be converted into ATP. So the net ATP is 2. Excellent. Well done. That's very good, man. This is shall come from lady. Well done. All right, good. Now, yes, let me listen to you.
How was the in the 5 ATP that we added to the 2?
Yeah, so here, what you are saying, this one here, because oxygen is there, when it gets, it will go all the way to electron transport. Okay, so when you get to the electron transport chain, they will convert this one into energy, into ATP. And when they convert this one into ATP, remember this one gives us what? ATP of equal to what? 2.5. Equivalent of this in when this one gives its electron, okay, at the electron transport chain, it gives energy equivalent to what? 2.5 ATP. But until it gets there, that ATP will not be, you won't get it. You understand what I'm saying? Do you understand what I'm saying here?
Yes, sir.
If it doesn't get there, and if there's no oxygen, even there, this process will not even occur. If there's no oxygen, this one will not occur. This one will not occur. This one will not occur. For this one to even occur. All right. So this one will only, all this process here will only occur from here going to occur only when what? Okay, only when, okay, all the one here, in presence of what? In presence of oxygen. So if there's no oxygen, all the damn process will not occur. And if it doesn't occur, this one here will not even get to the what? This one will not get to the electron transport. Okay, it will not get there. To not get for the electron transport to occur, so that its energy will be released. Do you get my argument? Please, do you get my argument?
Yes, dog.
Oh no, you don't sound convincing. I'm listening. Let's take it this way, that you have gold, but you have to sell the gold to get the money. Now, but the gold or the land is there. The land is there. You have gold. Ajiman, please. The father said the land is there. Don't go and do galamsey. You have one. So let me add disclaimer to it. I don't trust him. So let's say you have gold in front of your house.
Do you do galamsey in Tema?
So
do we do galamsey in Tema?
It's okay. So
galamsey has no respect for any place. So
yeah, thank you. So you have the gold there, but until the gold has been refined and sold, okay, it is not money. Until it has been extracted, refined, so there's no money. So here, yes, you know there's gold in front there, but that doesn't make any meaning. You know there's gold in front of your house, yes, but until it is being dug and then processed, refined, and then you get a pure gold, it is still nothing. All right. So this is how, and that one can only happen only when here. So this one here. So if glycolysis, if the work ends up in glycolysis, it's only 2 net ATP that will be produced. This one will not be processed at the what? At the electron transport chain for you to extract that 2 ATP from it, the 2.5 ATP from it. Okay. All right. Are you there? Good. Now, so this is all that you need to know. Okay, I've explained this one all to you. Okay, the passing there, chemiosmosis, and everything here. Now, okay, everything there is what I've explained here. Okay, as the pump, as the electron goes there like that, as it's going through this, look at that. This is the turbine. As it goes through the turbine, then it turns to what? NADH. Okay, ADP, that's adenosine diphosphate. Okay, and then they'll add inorganic phosphate to it to get our ATP as the hydrogen ion is being pumped through. Okay, like that, simple like that. Okay, and then so there's more or less the turbine. So the ATP synthase is equivalent to the turbine. So as it spins, the hydrogen ion going through them, then we are generating our ATP. Okay, that's very, very simple process. Now, the last one here, okay, So sometimes we do some simple calculation here. We call something respiration coefficient. So respiration coefficient is what? How do we calculate it? We calculate the what? As a ratio of what? Carbon dioxide produced over what? Over oxygen consumed. Okay, the ratio of carbon dioxide produced over oxygen consumed. Okay, all right, so that's how we talked about what respiration what coefficient. Okay, so it indicates what, so it helps us indicate, talks about cellular respiration and then the metabolic process. Okay, all right, so that is a what, so every substrate, okay, and it's what, it's respiration coefficient. You can see that carbohydrate has 1, so glucose like that. You can see that all the carbon dioxide that entered, all the glucose that entered came out as carbon dioxide. All of them, not even single one. All of them, all of them. If you can remember, 2 came out during the what? Formation of the what? 2 came out during the formation of the acetyl-CoA. All right, and then the other 4 came out in what? In the Krebs cycle. That from the what? From the isocitrate to the this thing. From the isocitrate to all the way to the I think the succinyl CoA. Within that stretch, all the carbon dioxide disappeared. Okay, all right. So here, this is the what is the so amount of oxygen you consumed and how much carbon dioxide produced. And you can see that. So here, this one tells you that carbohydrates, okay, almost everything that entered, okay, it's actually consumed typically almost everything there. Protein will give 0.9, and then fat 0.7, fat associated with carbohydrate 0.5, and fat associated with that. Okay, so you can see clearly here that you can see the carbohydrate is given almost close to 1, glucose specifically is given, okay, exactly what 1, okay, like all the amount of carbon dioxide produced over the what oxygen consumed. Okay, so we use this one to assess the what the metabolism, okay, of that is occurring within cells. Okay, all right, great. Now, if I'm not going to worry you, please let me steal a little bit of your time to explain this particular these 2 slides, and then we call it a day. But where we are, if I pause now, wouldn't feel, I would want us to just go through this one quickly, and then we are done for the day. Okay, all right, don't worry yourself about this. Yeah, hello.
Yeah, Doc, just a minute to
okay. Yes, please.
I had the question earlier on.
Oh, please go ahead, please go ahead.
Yeah, but for now, in the course of the explanation, I think the question is answered.
Okay,
the breaking of the glucose into carbon dioxide.
Yes, please.
The first stage, the first 2 that are released, at what stage are they released?
That is between the pyruvate to acetyl-CoA, the intermediate process. So remember the intermediate process during the formation of the acetyl-CoA, during the formation of acetyl-CoA.
Okay.
Formation of the acetyl-CoA. All right, here, have you seen during this process, 2 carbon dioxide molecules go away. Okay, so because you have the 2 pyruvate, each of them will undergo decarboxylation, and the decarboxylation they remove the carbon in the form of carbon dioxide. And during the Krebs cycle, each Krebs cycle, yes, and then each Krebs cycle will move 2, and there are 2 of the Krebs cycles, so they will move all the 4. All right, thank you.
All right, thank you so much, Doc.
All right, good. So this last part quickly, this one, I know your time is really gone, but in fact, I don't want to go and come back and do this one because so that it will stick in your head quickly. All right. Now this side is very simple. Let me just take a blank page and then do it quickly. Okay. Let me take a blank page. This is not difficult at all. It's not really difficult at all. Now, from what I said earlier on, okay, we know that after glycolysis, okay, what happens after glycolysis? Okay, the fate of the word pyruvate, okay, all right, after glycolysis, we produce pyruvate, okay, this pyruvate is what? 3 carbon molecule, okay, 3 carbon molecule. All right, now this 3 carbon molecule we are talking about, how does it work? Now, when oxygen is there, when oxygen is present, when oxygen is present, okay, what will happen? Acetyl CoA formation, followed by what? Krebs cycle. Okay, Krebs cycle, followed by the word electron transport chain, like that, when oxygen is present. Okay, great. Now the question is, when there is no oxygen, what will happen? All right, so that's the $1 million question. Now the question on the board is, now we see that oxygen, what about you finish and then there is no oxygen, of which, let's take an athlete running and he has exhausted all the oxygen in his muscles. What is going to happen? So now the pyruvate is here. My pyruvate is here. Pyruvate is here. Two of them. Two pyruvates here. Okay. Beautiful. Three carbon molecules here. Like that. They are done. No oxygen. No O2. Now what is going to happen? Good. Now do you know what? If there is no oxygen present, what will happen? What will happen is we call it what? Fermentation. Fermentation is going to occur. So at the end of glycolysis, okay, glycolysis, if there is no oxygen present for acetyl CoA formation, Krebs cycle, and electron transport to go on, what will happen? Fermentation will occur. But why is fermentation occurring? And let me tell you why. The main reason, now before that, what is the product of glycolysis? What is the product of glycolysis? The product of glycolysis is this. Now, glycolysis requires what? You see, glucose plus what? NAD plus. Do you remember? Do you remember that? Do you remember this one?
Yes, Doc.
Good. It's going to give us what? It's going to give us the pyruvate. Okay, the pyruvate. Okay, 2 pyruvate. Okay, plus what? Plus 2. So let me add my ATP here. 2 ATP. 2 ATP there. Okay, 2 pyruvate plus my what? My 2 NADDH plus my 4 ATP. Excellent. So this is what we get in glycolysis. Beautiful. Now that I've gotten this one in glycolysis, so you can see that for glycolysis to occur, you need this one. You need this. The glucose could be there, but if these two things are not there, glycolysis, alpha ATP, usually they're always in the cell. But if this one is not there, glycolysis is not going to occur. We need the what? The NADP ion, NAD plus should be there, because during glycolysis it's oxidation, but it involves removal of hydrogen. And when hydrogen is removed, who is going to pick up the hydrogen? Is this guy, the guy here. This guy here is the one who is going to pick up the hydrogen. So if this guy is not there, glycolysis is not there. But glycolysis, even though it's a small process, it at least produces what? It produces a net ATP of what? Net what? Net of ATP of what? Net 2 ATP. 2 ATP. So it's not bad. It's not bad at all. You understand that? It's not bad at all. It's giving something. All right? It's not bad at all. is giving net ATP of 2. So it's not bad. So for if you take something like a red blood cell doesn't contain mitochondria. So which means that because it doesn't contain mitochondria, that one will only undergo just anaerobic respiration, which is solely glycolysis, and produce net ATP of what? 2. So some people, even though this may be small, but some people need it. Okay, like red blood cell like this, it needs this one badly. Okay, good. So if oxygen is present, we are fine. Everything will go. Large amounts of what? ATP will be produced. Large amounts of ATP will be produced. But when oxygen is not there, what is going to happen? Fermentation will occur. The main reason fermentation is going to occur is to generate NAD plus. Are you with me?
Yes, Doc.
So when NAD plus is generated, which process can occur again? Which process can occur again?
It's glycolysis.
Glycolysis.
Good. Good. So now you have glucose in your muscles. You are running, but you don't have oxygen to breathe. You are doing marathon. So no, you are running, so not much oxygen is going into your muscles. Okay, so the muscles have been denied of oxygen, but the muscles should still produce ATP. If it doesn't have ATP in there, in fact, the muscle will stop functioning. If a cell that doesn't contain ATP will perish. So at least, even though you are not having oxygen in the muscle for this one to go, for these 3 steps to continue to produce large amounts of ATP, what is going to happen is that fermentation will rather occur so that it will generate the NADP. When NADP is generated, the glycolysis, okay, glucose can react with this, okay, in place of what? 2 ATP, okay, so that you get what? You get this particular process all over. Have you seen it? So fermentation, the main reason fermentation occurs is to generate this, okay, so that the glycolysis can go on, all right? But this fermentation, we have 2 types. What are they? There are 2 types of fermentation. This fermentation, we have 2 types. We have something we call lactic acid fermentation, and then we have alcoholic fermentation. All right, so we have lactic acid fermentation and alcoholic fermentation. Okay, lactic acid fermentation and alcoholic fermentation. Now let's see, when it comes to the lactic acid fermentation occurs in animal and, okay, lactic acid fermentation, let me spell the lactic acid here. Lactic acid fermentation, okay, fermentation occurs in what? Occurs in animal cells and what? And bacteria cells. All right, lactic acid fermentation occurs in what? Animal cells and bacteria cells. What does lactic acid fermentation result? Lactic acid fermentation, the pyruvate, okay, pyruvate, which said 3 pyruvate, which is a 3 carbon molecule here, will be converted into lactic acid, which is also 3 carbon molecule. That is all. So this is the lactic acid fermentation. In the lactic acid fermentation, what is occurring is that the word. So this one occurs in what? In the word, in the animal cell and bacteria cell. So for instance, okay, when I am running and I don't have enough oxygen in my muscles, okay, lactic acid will build up in my muscles. The buildup of the lactic acid in my muscle, that causes the muscles to become fatigued. Okay, that is why people go for long distance, you see that they come and then the muscle is, or when you walk too fast, that's the reason that the muscle becomes so fatigued. When you walk too fast, the muscles become what? They become fatigued. The reason they become fatigued is that there won't be much oxygen in the muscle. As you're walking too fast, you deplete the oxygen in the muscles. And when you deplete the oxygen in the muscles, then lactic acid is what? Produced. And this lactic acid is what causes the muscles to become what? Fatigue. So don't forget that the pyruvate is changed into lactic acid. This one occurs in muscle cell as well as the what? As well as the bacteria cell. Okay. Now the bacteria cell, let me give you one bad boy example. In the vagina of women, okay, in the vagina of women, there is this bacteria there. We call them lactobacillus. Okay. So we have lactobacillus, okay, and this lactobacillus, what it does is that, okay, this is so in the genital tract of women, okay, we have this bacteria there, okay, so they produce lactic acid, okay, so in the vagina like that, in the lactic acid in the vagina, okay, all right, so that is why, so the female genital tract, okay, tend to be acidic. All right. The reason is that we have these bacteria. Okay. Example is what? Lactobacillus. Okay. Bacillus. What they are doing is that they produce what? Lactic acid. Okay. In the vagina. All right. I quite remember one of my students after one class like that was telling the lady that one of the girls in the class that, do you want me to check your lactobacillus? I say, hey. That's a very subtle way of saying things. You know what I'm saying? Okay. All right. So lactobacillus. All right. So you have those lactobacillus. Okay. Bacteria. So these bacteria, there are plenty of them in the vagina. Plenty of them. They are good bacteria. They keep the vagina healthy. All right. By producing this acid. All right. That is why all ladies. Okay, that is why all ladies, women in their menopause. Okay, menopausal women. Okay, the menopausal women usually this particular bacteria in the vagina. You need this sex hormone. We call it what? Estrogen, the female hormone, estrogen. Yeah. You need the female hormone to keep them. Okay, this is the female hormone, the estrogen, female hormone to keep them there. All right. So there are a lot of them. I know even about 6 or so. They have very nice name. After class, you can Google for them and you see they have beautiful name. Lactobacillus crispatu, a lot of very beautiful names. I don't know who named them. They have beautiful name, but it's worth it. I mean, women are very precious. Ladies are very precious, so that's fine. So this lactobacillus, they are there in the vagina and they are producing the lactic acid to keep the vagina healthy. Okay. Now for menopausal women like that, what happens is that this estrogen tends to they need this estrogen to keep this lactobacillus there. When your estrogen level goes down, this lactobacillus, they tend to reduce in number. And when they reduce in number, you can get a lot of infections in there. The one infection, when this bacteria, when they reduce in number, there's this infection people get. We call it bacteria vaginosis. Vaginosis. Okay. So this one, like when this lactobacillus in the vagina, when they reduce in number, there will be bad bacteria. We have some bacteria in the vagina that are very bad. Okay. Yeah, that are very bad. Okay. Let me see. I have example in mind. So bad one, like we have this one like this one. Let me reduce the size here a little bit. Oh, look at me. Okay, we have some bad bacteria. Okay, we have some bad bacteria in the vagina. Okay, let's see. I can reduce the size more so that. So we have some bad bacteria in the vagina. Example is what? Okay, Provetella. Provetella is one bad bacteria in the vagina. Another bad bacteria in the vagina. Provetella. And then we have this one. I love this one. This one is called atopobium. You know, honestly, if I were naming this one, I would have named this one. I would have put A here, but it's a white one who named it called atopobium. Okay, you understand that? Ajima, you get it right. If I'm the one, yeah. If I'm the one naming this one, I would have put A here, but I don't know. So we have the atopobium, and then we have this one. This one is very common. This one is very common with our women. Gardnerella. Okay, like that. This one. So these are bad bacteria in the vagina. And this bad bacteria in the vagina, what happens is that when the good bacteria in the vagina, which one? This is this one, the lactobacillus, when they reduce in number, okay, this bad one will increase in number. And when this one increase in number, they cause this one, they cause the disease. Okay, and research has shown that bacterial vaginosis is more common in black women than in white women. The reason, I don't know. Okay, well, maybe black women are much more endowed than white women. Maybe that could be the reason, but the exact reason I don't know. Okay, my best guess is that black women are much more endowed than the white women. Okay. So this bacteria you saw here, so this lactic acid is very important. We have a lot of bacteria like the lactobacillus. That's where they live. They live in the vagina. That's where they live. They are producing lactic acid to keep the vagina healthy. And I said that for women, this acid is very important. You need this female hormone, estrogen, to keep them there. That's why we advise women. Those vagina washes and stuff like that, they are no good. Those vagina washes could kill the lactobacillus. We could kill it. There are lots of them. There are about 8. I know about 8 or so. Okay, all right. So there are a lot of them. Okay, and when their number actually the number goes down, the Provetella, atopobium, and Gardnerella, they will increase in number, and they cause this one. Okay, they cause the bacterial vaginosis. Okay, all right, great. And then let me write something you will see. And then I can't say it because for respect of women, I can't say it. So let me type it.
You see, some things are for men.
What I'm typing is for men.
No, I'm saying some things are, meaning the women, they can't compare themselves to us. But please, look, I have a question.
Hey, please wait.
No.
After this one should be backdoor question. We have still plan to. This one is because of the lactic acid, and for you to understand, that's how I've gone this far. So please, it's a good one question.
Yeah, it's about the fermentation process that occurs in the absence of oxygen.
Are you sure?
I wanted to ask that question, but as you were proceeding, I realized you were giving some.
Okay, so let's see. Let me reduce this size at this point here. All right. So as I said, so this disease, this is what will happen. Okay, have you seen that something is very bad? It's very, very bad. Okay. Very bad. Okay. So our ladies, be careful. Those vagina wash and stuff like that. Okay. Well, okay, so good. So in the absence of oxygen, what happens? Fermentation occurs. There are 2 types of fermentation. We have the lactic acid fermentation and alcoholic fermentation. In lactic acid fermentation, the pyruvate is converted into lactic acid, which is also 3 carbon molecule. All right. And then because of our women here, I've deliberately added this one. Okay. And in the word, in the lactic acid fermentation occurs in what? Occurs occurs in the animals and what and bacteria cells. Okay, and that's changing of this one. And then I've taken the opportunity to talk about this one. Okay, all right, great. I'll delete this one from the slide when I'm sharing it. Okay, this is too bad boy. Okay, great. So I'm deleting it now, gentlemen. Please ask your question before I delete it. Yes.
Okay, please, Doc. Thank you so much for the insight. The question I wanted to ask is: so, in the view of this fermentation, which occurs in the absence of oxygen, are we then, or can it be related to anaerobic respiration when it comes
That's
to
anaerobic respiration? That's anaerobic.
Okay.
It's okay because there's no oxygen. The word anaerobic means no oxygen present.
Absence of oxygen. Okay.
That's it. The word anaerobic, that's the meaning of anaerobic. So what we are talking about here. So let me delete this one here. So everything we are talking about here, please let me. So everything we are talking about here is anaerobic. Anaerobic. Okay, anaerobic respiration. Anaerobic respiration. There's no oxygen. Okay, anaerobic respiration. Okay, the word anaerobic means absence of what? Oxygen. Okay, so this is this. So when oxygen is not present, what happens? So this anaerobic respiration. Okay, so at the end of glycolysis, if there's no oxygen, fermentation will occur. There are two types of fermentation. We have the lactic acid fermentation, alcoholic fermentation. In lactic acid fermentation, they occur in what? In animal cells as well as what? Bacteria cells. Okay, in the lactic acid, what happens? The pyruvate is converted into what? Into lactic acid. So that's why it is called lactic acid fermentation. And then my last statement on this side is the word is the alcoholic fermentation. Okay, alcoholic fermentation here. So let me copy this one, put it here. Alcoholic fermentation. The alcoholic fermentation, what is going to happen? Then the pyruvate will be converted into what? Into ethanol. Will be converted into ethanol. Ethanol, which is what? Which is CH2H5. Okay. Oh, I wish I could H5 OH. Okay. All right. Am I getting anything like here? So 2H here. Then please, I hope you know my numbers are subscript, right? I hope you know my number. All my numbers here are subscript.
Yes,
hello.
please.
PowerPoint doesn't have the symbol for me to that option for me to make it subscript. So all my numbers, this numbers are subscript. So you can see there are two carbons there, and then what else? Plus carbon dioxide. Carbon dioxide. That is all. So this one occurs in what? It occurs in fungi, and plants. So that's how we get the abe. The abensa.
Ajingo, are you there? Ajingo.
Now I'm getting to where you want to be. Yes.
The password of Ajingo.
Ajingo. Finally, I've entered the class.
Ajingo is a man of God. He don't take
finally, I've gotten to where you want to be. All right.
The password of Ajingo.
So this one here is why we are calling it what? Alcoholic fermentation. But in each of the cases, after the production of this, what substance is produced? NAD+. Okay, this one, don't forget that NAD+ is released. And this NAD+ is very important so that the glucose is present, still the glycolysis will occur, and then still energy will be produced. So even when an athlete is running and there is no oxygen in the person's muscles, still the muscles wouldn't perish. What will happen? The glycolysis glycolysis will occur to produce net ATP of what? 2. Okay, so that the reaction can still, so that energy can still be produced. But the main reason fermentation generates this. All right, thank you. Please, any questions so far? Don't forget that in the case of alcoholic fermentation, ethanol is carbon two carbons. Have you seen? So the third carbon is what is being released as well, carbon dioxide. In the case of lactic acid fermentation, carbon dioxide is not produced. In lactic acid fermentation, carbon dioxide is not produced. Have you seen here? Here, no carbon dioxide is produced. Here, in the case of alcoholic fermentation, 2 carbon, which is the ethanol. 2 carbon here is ethanol here. All right. This 2 carbon ethanol, and then carbon dioxide is produced. And this one occurs in what? Fungi and plants. Okay. As well as the release of the most important reagents here, so that it will make this one available for glycolysis to continue. Please, any questions so far? So in alcoholic fermentation, ethanol and carbon dioxide are produced. In lactic acid fermentation, only lactic acid is produced. Okay, all right, great. Please, any question? So on this note,
please, Doc.
Yes, go ahead.
I have some question.
Please go ahead.
Along the line, I learned
please,
that
it should be good boy question. It should be good boy question.
Yeah, I learned that with this anaerobic respiration.
Yes, please.
Because of the production of the lactic acid in the muscles.
Yes.
When that happens, at times it causes some pains.
Yes, fatigue.
When we are working.
That's why. That's so the accumulation. Good. So here. Okay, so let me duplicate this slide. Let me duplicate this one. Let me move this one double so that's it. Okay, let me duplicate this one and clear the top. Okay, clear here. Clear the top one so that I can. Okay, and let me write anaerobic on top here. Anaerobic here. Okay.
There's a type of aerobic that most athletes use when they are running.
Yes.
So that is why I
don't have to run too fast.
So taxed.
So if I'm going long distance, what it means is that when you start, you don't have to run too fast. Because when you run too fast, initially, if you run too fast, what will happen is that you deplete the oxygen in your muscles quickly. And when you deplete the oxygen in the muscles quickly, what will happen? So let me kick this one down. What will happen is that, okay, in the case of animal, let's say in your muscle, what is going to happen? There will be accumulation of what? Accumulation of what? Of lactic acid in the muscles. Okay, specifically skeletal muscles. Skeletal muscles. Okay, and this what causes, okay, and cramps. This causes the what? Causes the, I won't use cramps if you're a bad boy. Okay. This
That's exactly what happened. I'm a sportsman, so I know what I'm talking about.
I say I will use scribes.
If you don't use
I'm a bad boy. Keep quiet. Keep quiet.
You want to?
What causes fatigue in the muscles?
Okay, so this is I will answer. Okay, the lactic acid accumulation. This is what causes the fatigue. This one. Okay, so the accumulation of the weight. So when the athlete is running, okay, so when they are training, you tell them that, oh, do you know what? Train such a way that when you get a certain pace, don't move too fast so that you deplete your muscles with oxygen. Because the moment you deplete your muscle of oxygen, the pyruvate will be converted. Instead of, if there is oxygen in your muscle, remember the pyruvate will be converted into what? Into acetyl-CoA. And in that case, the Krebs cycle will be going on. The Krebs cycle will go on and the electron transport will go on. But if there is no oxygen, if let's say you run too fast to deplete the oxygen in your muscles, in that case, what is going to happen? What is going to happen is that they will be going to what? Once you deplete the oxygen there, once the oxygen is depleted, the pyruvate will be converted into lactic acid. Okay, into lactic acid. And when they convert into lactic acid, the lactic acid will begin to accumulate in your muscles, and that will cause the muscles to become fatigued. Okay, the muscle will become fatigued. So we tell the athlete that when you start, don't move too fast. Get a certain rhythm where, so we call it move at a speed so that you don't hit the anaerobic threshold. That's what we advise athletes. Move, okay, so that you keep yourself below. Move and keep yourself. We say that run such a way that you keep yourself, okay, below anaerobic threshold. Anaerobic threshold. That's what we advise athletes. Anaerobic threshold. Okay, so don't run too fast. Okay, if you run too fast, you quickly deplete the oxygen in your muscles and you become so fatigued early. Okay, and you can't run. But so just keep your pace so that you still can have some amount of oxygen in your muscles. Okay, all right. Below anaerobic threshold. That's what we advise the athletes. Okay, so when you accumulate the lactic acid, okay, in the muscles, then it will cause the muscles to become fatigued. So at the end of the race, what is going to happen is that this lactic acid will be transported to the liver. To the liver. In the animal cell, in like those of us here, okay, it will be transported to the liver and then it will be converted. Okay, it will be converted. Like, even while you are running, that's what happens. Not even at the end of the race. While you are running, the lactic acid will be transported into the liver. Okay, the liver. And in the liver, it will be converted back to glucose. Okay, back to glucose. By a certain process, we call it gluconeogenesis. Gluconeogenesis. All right. So when it goes back to the glucose, they will change it. When it gets to the lactic acid, okay, that process we use a certain, we call it Kobe cycle. Okay, that's beyond this class. I don't want to worry your head. Okay, all right. The most important thing is that in the absence of oxygen for animals and bacteria cells, lactic acid fermentation occurs. And lactic acid fermentation is a conversion of pyruvate into lactic acid and then NAD plus. Please here, no carbon dioxide is released. But for alcoholic fermentation,
ethanol and alcohol is produced.
Ethanol, which is the alcohol. Alcohol is the ethanol, please. There's ethanol here is the alcohol. Okay, the ethanol and carbon dioxide are produced. So on this note, I would say I'm done. What is left is photosynthesis. Ajiman, discuss with your brothers and sisters, and then just give me time. Give me about 2 different times, and let me pick one. Okay, that will be. Well, I'm sure if you just give me one, probability that I'll be very busy will be high. So, but that the day shouldn't be. The day shouldn't be
Monday and Wednesday.
Monday and Thursday, rather.
Thursday, okay.
Right. I mean, okay, yeah. So, So you just give me 2 days and let me pick one evening. Let's do the photosynthesis, and that photosynthesis 2 hours we are done. And I'm so happy you've understood this one very well. I know I've taken a lot of your time. Thank you very much. May God richly bless you. Have the best of the day, and please do have fun. Okay. All right. Thank you.
Bye.
Please, Doc, we thank you so much for the insight. You're most grateful.
Thank you very much. Now, if you want bad boy question, now you can ask bad door. My ladies, if you want to ask a question, please, that one back door. Okay. Ajiman, I won't take a question from you, please. Okay. I know you.
There's a question in the chat box.
Yeah. Oh, there's a question in the chat box. Let me go there quickly to look at it. If it's a man, let's see. No, there is no question. Okay.
There's a man there.
From glycolysis, formation category, website, selection transfer. View occurs. Oh yeah, so it occurs steps in steps. Yes, you are right. That's it.
Okay.
It's okay. It's stepwise. It's stepwise. You're right. So does the lactic acid fermentation occur in both plants and animals?
No.
No. Lactic acid fermentation will occur only in what? Animals. But alcoholic fermentation will occur in what? So lactic acid will occur in animals and okay, so lactic what? Lactic acid fermentation occurs in what? In bacteria and animal animal cells. Okay, and then while the alcoholic word um alcoholic fermentation occurs in what? Plant and fungal cells.
Okay.
All right, good. Okay, good. So I think on this note we can call it a day. Okay, so I'm going to organize my quiz now, and then if you have any question, please, my WhatsApp number is there. Those of you who don't have it, 77144915. But I'll be in a quiz room, so it will take time for me to respond though. Okay, maybe for that. Okay, so maybe by the end of 2 o'clock, I'll definitely respond if I have any question for you. Right, good. So thank you very much, and then enjoy your day. God bless
Thank
you.
you, Doc.
Thank
God
you.
bless you too, Doc.
Yes, thank
Doc,
you.
may
Have
God
a
bless
nice
you.
day.
You're more
Amen.
grateful.
Amen.
We'll be calling you. Hmm. I'm offended.
Hello, Ajingo. Hello, Cost Rep.
Yes, I'm here.